Ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes.
Can you hear me? Still.
Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session.
I don't have the, I can't hear you all. I'm just wondering where.
Good morning, everyone, and welcome to our 2023 NanoDay Symposium. Continuous manufacturing of nanomaterials. Good morning, good afternoon, or good night, depending on where you may be. It is so exciting to see so many of you online with us here today, and we all certainly appreciate your commitment in attending this event and meeting any scheduling challenges that you may have had. My name is Brenda Stoddart. And let me change my screen a little here so I could see me better, see my slides better. My name is Brenda Stoddart, and I'm with the Small Business and Industry Assistance Program, SBIA, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communications. It is my great pleasure, along with my colleagues, to be with you here today for what is our second consecutive NanoDay Symposium. This event features subject matter experts from FDA's National Center for Toxicological Research, Office of the Commissioner, Office of Pharmaceutical Quality, and academia. We entreat you to make the most of this experience, attend all the sessions that interest you, learn all that you can, submit your questions during the available Q&A sessions, and network in the chat pod. In addition to the Adobe Connect platform, we are offering live YouTube streaming, so please go ahead and invite your colleagues. However, please note that only those attendees who register and attend via Adobe Connect and log into Room 1 will be able to submit questions to our speakers. Now, as we set up for this poll, I know many of you have been with SBIA for quite some time, and perhaps even going back as far as 2010 when we first started, although I dare say that many of you are also first-time attendees. as a special welcome, well, not as a special welcome, but a special welcome to you as we work towards expanding our audience base and increasing our portfolio of SBIA events. So to break the ice and get us thinking, I have the following question. Please participate if you can. And I see some of you have already started. That is very good. Thank you. And the question is, how many SBIA events, not necessarily conferences, we do a lot of webinars as well, but how many SBIA events have you attended? And your choices are, this is my first one, 2 to 5, 6 to 10, more than 10, most since 2010. And let's see, Ray, can you take a snapshot of this for me, please? Let's see if the numbers are evening out here. Anyone else? Okay, it looks like 36%, the majority of the audience. This is your first event. Thank you so very much and welcome. It's a great event to be here with us. And 2% attended almost since started. Welcome back, and thank you for your loyalty. Thank you all for participating. Okay, on this next slide here, as of this morning, we had actually over 1,700 registrants representing 75 countries. Of these, 46% have identified as small businesses, and of those, over 31% report having 1 to 9 employees. And these stats continue to amaze us and are extremely important to us as they confirm that we are indeed reaching the small business sector of our audience, and they also validate our mission and the very existence of the program. So thank you for, helping to make the program continue and its longevity. This here is a snapshot of a geo map for today's event. And this map is always quite invigorating to us here at FDA and our planning committee, for it provides a very powerful visual as to the impact and extent of our outreach. And you can actually click on this image, which will take you to your browser, and you can zoom in and have a closer look at our global participation. I think that's always a fun thing to do first thing in the morning. I invite you to try it, zoom in on your geographic area, and see if anyone else is attending from your neighborhood. This slide provides the QR codes for social media avenues, and you can scan them with your phones and be taken to the appropriate platform. They will also be advertised across the screen banner during the breaks if you miss it now. And let's turn to some logistics. In our ongoing environment, this event is again 100% virtual. Over the course of the day's event, we'll have a total of 10 speakers and panelists, not to mention our organizers and technical staff, all working from our own locations. As such, there may be some internet issues, but please bear with us. And if in fact we encounter those, we will try to get them resolved as quickly as possible. And speaking of tech issues, here is the link for a one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. That's the same page that you used this morning for your online access to the event. So please bookmark that page as you already did over the course of the day. If you have any technical questions about this symposium, please click there first and you will most likely find your answer. I can't stress how much we invite you to submit your questions to us so that we can have a vibrant and productive Q&A segment, which is the lifeblood of these events. This is a learning opportunity, and I encourage you to take full advantage of the expertise we offer here. We will answer as many questions as time allows, and when you do submit your question, please identify that individual presenter by prefacing your question with the speaker's name as shown here. Just one reminder, we are not able to answer very specific individual application questions. Also, during each presentation, there is a link on the right side of the pod where you see the speaker's headshot and it says download slides. And note that this link is only active during the real-time presentation. So if you were to try to get this on YouTube after the fact, you would not be able to download the slides at that time. There is also a link to a speaker evaluation, We encourage you to take the time to complete the evaluation, as your feedback is much appreciated by the individual speakers. In addition to the speaker evaluation, we have a final evaluation. Upon completion of this survey or evaluation, whichever term you like to use, an attendee can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. The trick is that the certificate must be downloaded immediately after completing the survey, otherwise that opportunity is lost. Now, even if you don't want or need a certificate, please take a few minutes to complete a conference evaluation, as your feedback will help guide the design of our future events. And note that both the evaluation and certificate are available for only 2 weeks post-event, which means that for this symposium, the deadline date is October 25th. The survey link will be posted on our one-stop shop page this afternoon. As a reminder, all the sessions are being recorded. It will take us a week or so to process the recordings, but you'll soon be able to access them from SBIA's homepage. And I hope that you have already bookmarked our homepage, and that is fda.gov/CDER SBIA. That's all one word, CDER SBIA. In the interim, the YouTube link for today's recording will be posted on the One Stop Shop webpage at the end of the day. And to simplify things, SBIA will send one email to all the Adobe attendees later today post-event. And that information will also have the conference evaluation link in case you miss it during the event today. Okay, so let's take a look and highlight some of our upcoming events. On October 16th, that's next week, we'll have our webinar on the BSUFA 3 Regulatory Science Pilot Program. This is a two-part meeting on October 16th, which is the virtual webinar. And then on October 16th, we'll have the in-person meeting. Detailed information is posted on our website. Then on October 31 through November the 1st, we'll be hosting our biannual Pharmaceutical Quality Symposium 2023, where we will explore topics related to pharmaceutical quality regulation, supply chains, and advanced manufacturing technologies. On November the 16th, we'll have our Common Issues with SEND data submitted for safety pharmacology studies. And from the information provided in this webinar, industry will be able to apply this knowledge to update their systems to avoid these errors in the future, and so increase usability of the SEND data sets for regulatory reviewers. We still have quite a few more 2023 SBIA in the pipeline, and more information will be available on our homepage seen here, again, fda.gov/CDER SBIA. And we also invite you to visit our CDER Learn webpage, where you can link to SBIA tutorials along with other educational resources, many of which offer healthcare's continuing education credits. And that website, of course, is fda.gov/CDER Learn, all one word. What you see here is a short video, which will give you an idea of the SBIA resources available to you. We won't play it now, but it will be played during the lunch breaks. And at this point, I would like to acknowledge all the individuals, well, many of the individuals instrumental in making this event possible. Many names are listed here, but so many more deserve credit as well. Most of all, our heartfelt gratitude to you who took the time out to attend this event, regardless of the inconvenience of various time zones. And we do not take this lightly, for without you, SBIA wouldn't exist, really, you know, and definitely without you, the symposium and similar events would not be the success that we anticipate it will be. A very special recognition to my SBIA colleague, Captain Ray Ford, who will be your host over the next 2 days. And now I'm sure you're ready for us to launch fully into today's agenda. And as a reminder, all our speaker bios are available on the One Stop Shop website. But for now, I am honored to introduce Dr. Anil Patri, who will officially welcome us all. Dr. Anil Patri leads FDA's Nanotechnology Task Force and oversees collaborative regulatory science research at the Nanocore. His lab developed 7 international standards in nanotechnology through ASTM International in the past 4 years and contributed to many other documentary standards. Dr. Patri serves on the Nanoscale Science, Engineering, and Technology Subcommittee on behalf of FDA for interagency coordination and leads the US-EU communities of research and characterization. Prior to joining FDA in 2014, Dr. Patri served as the Deputy Director of the Nanotechnology Characterization Laboratory at the Frederick National Laboratories for Cancer Research and as a guest scientist at the National Institute of Standards and Technology. Let us please welcome Dr. Anil Patri.
Thank you very much, Brenda. I echo your comments about the many that contributed to this symposium. Good morning, everyone, and thank you for joining us today. My name is Anil Patri, and I would like to welcome you all for this year's FDA NanoDay Symposium on continuous manufacturing of nanomaterials. I would like to thank Brenda Stoddart and CDER SBIA program for assisting us with the NanoDay Symposium every year. Some of you may have attended the last year's symposium, which is highly successful. And for this year, especially, I thank Dr. Xiaoming Xu for leading this year's symposium. It continues to be an exciting time for nanotechnology with last week's 2 Nobel Prize nods for research on mRNA COVID vaccines for physiology and medicine, and for research and application of quantum dots in chemistry. Today's symposium is dedicated to the emerging continuous manufacturing of nanomaterials intended for medical products. Continuous manufacturing is in use for small molecule drugs for some time and only recently applied to the field of nanomaterial synthesis, especially for liposomes and lipid nanoparticles, and can be extended to others. FDA has funded this effort through broad agency announcement grants, and we have an excellent panel of speakers today, both from within FDA and those that are funded by the FDA, to present about the past, present, and future applications, and how this will potentially address challenges in drug shortages while assuring the quality products to benefit patients. I would like to now request Dr. Nemanje Bumpus, FDA Chief Scientist, to provide opening remarks. Dr. Bumpus joined FDA a little over a year ago with distinguished career in academia, more recently at Johns Hopkins University as a professor and chair of pharmacology and molecular science department. At FDA, she is spearheading the research, science, and innovation that are fundamental for our regulatory mission. Dr. Bumpus?
Welcome, everyone. I am Nemanje Bumpus, the FDA Chief Scientist. I would like to congratulate the Nanotechnology Task Force and the CDER Small Business Industry Assistance Program for organizing this NanoDay Symposium to showcase FDA's work on continuous manufacturing of nanomaterials. I would also like to thank them for the excellent work that they do throughout the year. The mission of the Nanotechnology Task Force is to leverage FDA scientific expertise and resources to advise agency and center leadership on critical and cross-cutting nanotechnology-related issues to support the development of safe and effective FDA-regulated products that may contain nanomaterials or otherwise involve the application of nanotechnology. This symposium serves that mission. FDA has been more actively involved in celebrating NanoDay over the last 4 years. In 2020, we celebrated it by releasing the Nanotechnology Task Force report. In 2021, we had an internal research symposium at FDA for NanoDay. In 2022, we had a public meeting to highlight the final guidance to industry from CDER and CDER on nanotechnology and drug products. Last year was very successful with over 3,500 virtual participants. This year, we wanted to highlight the emerging area of continuous manufacturing due to its importance in advanced manufacturing, a topic of interest within FDA and globally. Continuous manufacturing is an emerging technology that improves product quality, reduces waste, lowers production costs, and enhances manufacturing flexibility and agility. This symposium intends to share FDA's experience in this emerging area and hopes to inspire developers and researchers to adopt and realize the full potential of nanotechnology and continuous manufacturing. We're only starting the journey. From a research standpoint, significant work lies ahead to enable development and translation of therapeutic and vaccine products utilizing continuous manufacturing. Enjoy the symposium and happy Nano Day!
I would like to now introduce Xiaoming Xu, the Division Director for Office of Testing and Research, Office of the Office of Pharmaceutical Quality, CDER, FDA. Xiaoming also serves on the Nanotechnology Task Force as a member from CDER along with Olin. And he has been very active in both research at FDA in nanotechnology and leads an excellent research group. So he's the main organizer for this symposium. So I would like Xiaoming to say a few words before taking it over. Xiaoming?
Hello, everyone. I'm Xiaoming Xu, the co-chair of the 2023 FDA NanoDay Symposium. I want to start by recognizing my colleague, Dr. Anel Patry and Dr. Olin Stevens for the guidance and help over the last few months planning for this event. Together, also, we would like to thank our amazing staff at SBIA for organizing today's event. In the next few minutes, I want to briefly share with you our thought behind this event and introduce to you the topics that will be covered. This symposium is focused on the convergence of two interesting fields, nanotechnology and advanced manufacturing. It's a rapidly developing topic, particularly at the intercept of the two fields, to approval of nanotechnology products manufactured by continuous manufacturing. However, we hope that you can see the potential, maybe after today's event, and that we can all work together to push forward the field, and maybe one day to approve the first nanotechnology drug product manufactured by continuous manufacturing. Now, as you might also notice, the title of this slide includes the phrase, learning from the future. Isn't that strange, as we typically only look into the past to learn? Let me briefly explain it. As we have all experienced it, we tend to conceive time as linear and unidirectional, such as moving from the past to the present and to the future. We remember yesterday, we experience today, and we look forward to tomorrow. However, to plan or to prepare for the future, especially in uncertain times, sometimes we need to embrace a nonlinear conception of time. And this concept is coined by Dr. Peter Skoplik in his 2020 Harvard Business article named Learning from the Future. What this concept entails is that sometimes we need to step away from the traditional view of time and sequence of events to take a leap forward as well as backwards so that we can influence the future instead of just anticipating it. As he described in the article, sometimes we need to take a closer look of the trends in the present and jump years into the future and to describe a plausible world of future scenarios, then work backward to develop stories about how the future world may become, and then work forward again to develop strategies to realize that future. And this concept is widely used in the business field. Also, it's called strategic foresight, which is drawn from many successful cases from other industries. It prepares the organization to better weather the storm in particularly difficult times, which is often hard to predict, such as a COVID-19 pandemic. So I bring up this concept as a preface to the conference because I see parallels in scientific research. Maybe this view of the connection is easier to understand. What we view as today's success, especially in scientific breakthrough, is often a deliberate effort from the past. After all, scientific research takes long time and dedication to accomplish. For example, concept of nanotechnology is not new. Indeed, Dr. Benham identified the spontaneous formation of vesicles from phospholipids more than 6 decades ago. And since then, countless researchers had dreamed big and started experimenting with lipid vesicles to encapsulate various types of therapeutic agents to take advantage of this natural delivery platform. And you must have heard about the news last week of the Nobel Prize Award to Dr. Kathleen Carrico and Dr. Drew Weissman for their contributions to the messenger RNA research, which is a crucial tool in combating the COVID-19 pandemic. Big congratulations to them for such worthy recognition. If we think about it, the anticipation of the use of photogenic lipids to help with the delivery of RNAs has been perceived more than 2 decades ago. And today's success in COVID vaccine did not begin after the pandemic, but instead started much earlier. So, as we celebrate today's success, we cannot overlook the effort from the past, which was often a deliberate effort to change the future. Speaking of nanotechnology, I would like to remind everyone about the view at the agency about if and when do we consider FDA regulated product utilized nanotechnology. The first point to consider emphasizes on the dimension of the engineered nanomaterials, while the second point to consider emphasizes on the purpose and the impact on properties relevant for products' intended effect. Evidently, over half of FDA-approved drug products containing nanomaterials with dimension between 100 nanometers and 1000 nanometers. So when you hear the term nanomaterials, do not assume it is only for those that are less than 100 nanometers. Now let's go back to the theme about learning from the future. What we can imagine as the future that we can work towards today, especially in regulatory science research. Maybe it's making the nanotechnology product more accessible, even more robust against drug shortages. After all, many patients cannot afford to lose access to their life-saving drugs, such as oncology treatment or crucial vaccines. Maybe it's to prepare the industry and the regulatory agencies to be more agile so that we can more easily introduce or tolerate changes, whether it's to change a supplier of a raw material or changing location of the manufacturing. Maybe it's to better prepare for a future of more individualized treatment. For example, with so much progress made in the human genomics and understanding of individual risk factors, can we develop products targeting individual patients rather than a one-size-fits-all approach? Of course, we design a future where the nanomaterial drug product can become more affordable, so we need to bring forward paths to encourage the development of the generics. Lastly, patients need to trust that industry has developed a medicine that has been rigorously evaluated so that they have confidence about the use. Now, let's all think about a future where it is better than today and how we can get there. Speaking of trust, everyone deserves the confidence in their medicine, not just the one went through the clinical trials and approved by the FDA yesterday, but the ones that the patient will receive tomorrow in their hand. What allows them to have the confidence is the pharmaceutical quality, which ensures that the availability, safety, and efficacy of every dose is something we need to continue to improve. One way to improve the quality of drug product, and certainly it is an opportunity area for nanomaterials, is the use of continuous manufacturing. Similar to nanotechnology, continuous manufacturing is not a new concept. However, in pharmaceutical applications, it is a rather new application. We have one talk immediately following this one to provide a more detailed explanation and overview about FDA's experience so far on continuous manufacturing and what we can learn to apply to nanomaterials. To close my portion of the talk, I would like to introduce to you the presentation topics of today. you will first learn about our experience in developing and approving continuous manufacturing drug product, kind of what is a current trend. And then we will share with you our past effort invested in the area of continuous manufacturing of nanomaterials from 2 FDA grant awardees at University of Connecticut and Princeton as well as Purdue. We will follow that with some discussion about current regulatory challenges and opportunities, as well as ways that you can interact with the agency to help pushing forward the regulatory science research. Lastly, let's start taking action today so that we can all change tomorrow. And thank you very much, and I hope you will enjoy this symposium.
Thank you, Dr. Xu, for setting the stage for the remaining presentations. This is Ray Ford working with SBIA, and it's my pleasure to be your host for the remaining sessions. Moving into our first presentation on CDER's perspective on continuous manufacturing journey, past, present, and future. This will be provided by Dr. Thomas O'Connor, the Deputy Office Director in the Office of Testing and Research in OPQ. Next, we'll learn about continuous manufacturing platforms for lipid and polymer-based nanoparticle therapeutics from Dr. Antonio Costa, University of Connecticut. For a complete biography on each presenter today, please refer to our speaker biographies located on the event page for all event details adjacent to the event agenda. Please join me to welcome our first presenter, Dr. O'Connor.
Hello, my name is Thomas O'Connor, and I'm excited to join you today to talk about CDER's perspective on the continuous manufacturing journey. First off, I want to focus the talk on pharmaceutical quality. So that is really that every patient deserves confidence in their next dose of medicine. Pharmaceutical quality is really assuring the availability, safety, and efficacy of every dose. I want to start off today's talk by defining what do we mean by continuous manufacturing. Certainly, each individual unit operation in a manufacturing process could be continuous, where the material continuously flows in and flows out of the unit operation. But we're talking about continuous manufacturing, really focus on an integrated process that really consists of 2 or more unit operations that are connected together. And what that means is that there's less isolated material during the course of the processing because of this integration, and that drives more reliance on real-time data collection and decision-making. And FDA has identified, long identified, continuous manufacturing as a novel technology in the pharmaceutical industry, which may not be a fit for every drug, but can bring potential benefits related to increased efficiency. It usually results in more intensified processes that reduce the footprint of the process. And again, this increased data collection in real time to facilitate the control of the process improves the process control. And while at the start of this journey, those benefits were potential, we have illustrative examples where the companies have realized those benefits. So this is a report from one company that switched from a batch process to a CM process and reported significant reductions in operating costs and reductions in efficiency, so reducing their waste, and increasing both their cycle time for manufacturing as well as testing. Using more of that data from the actual process to speed up some of the end product testing. Not only does that translate to benefits that can be quantified in that respect, CM can also enable processes that just couldn't be safely conducted using batch manufacturing processes. This is one example reported by a company that used CM for a Grignard reaction, which results in and allows spontaneous heat release. So the continuous process was able to be safe and well controlled and really enabled this reaction to take place, which couldn't be safely executed in a batch reaction mode. From the FDA's perspective, not only are there potential benefits, for industry, but there are also potential benefits for patients, including, better quality medicine. This improved control can reduce, the amount of defects that are produced. Continuous manufacturing, usually involves less scale up because it's intensified processes, resultant processes that can be, you can kind of use the same rig for development as you do for commercial manufacturing because they're smaller and use less material, and that can really help speed the development and getting those products to market faster, especially if for these drugs that have accelerated clinical development timelines. By reducing quality issues can help prevent drug shortages. And then having more flexibility and agility in the manufacturing sector can help improve emergency preparedness. So having a manufacturer that can quickly scale up the amount of product being manufactured. So I just want to kind of take you through a little bit of a timeline to review some of the key highlights along this continuous manufacturing journey. It's labeled here, and I'm going to start off in 2014, not because 2014 is actually the beginning of the journey. That actually started much earlier when there was a number of investment in some of the science and actually the equipment that happened both in academia and industry that predates that by over the previous decade. But 2014 is really kind of a turning point, a tipping point, as you will. So at that year, we had our first international symposium on continuous manufacturing, really bringing together international regulators together for the first time. And we had the forming of CDER's Emerging Technology Program, which has really been the program that's really interfaced with companies looking to implement continuous manufacturing. 2015, big milestone, the first drug product approval that was made using continuous manufacturing, as well as two papers that published that really outlined some of the scientific thinking behind quality considerations. One was a joint industry regulator paper, and the other paper was from the FDA. In 2016, we even had the first approval of a product that was made by batch manufacturing, switching to a continuous manufacturing process. In 2017, we continue to see additional approvals in the drug product space, as well as finalizing the guidance for the emerging technology team, a key milestone for that program. 2018, we see the formation of ICH Q13, which is an international harmonized guidance focused on continuous manufacturing, so endorsement. So that's just the beginning of that process, but there's a key milestone, leading to international harmonization. 2019, again, you see additional approval, but also FDA publishes draft guidance on quality considerations for continuous manufacturing. It's important to note that that guidance is going to be more limited in scope. It's going to be focused on drug product and solid oral dosage forms, which really form the basis of our experience up to that point. In 2020, we start seeing approvals in more expanded applications. So we have first approval for continuous drug substance process, first approval for a semi-continuous biotechnology process, even for sterile drug products, these semi-continuous processes. So we're starting to see continuous manufacturing being adopted by for other types of drug products. And then 2021, a milestone was reached, which is the graduation of the continuous direct compression from the Emerging Technology Program. That really signifies that we had enough, knowledge gained over the past, 7 years that those type of applications can actually move from, the Emerging Technology Program and can be really handled, through the normal assessment channels. That industry had enough experience implementing these type of processes. 22, first approval of a CM process at a CMO facility. So not only these processes, commercially available at innovator companies, but actually made available to others at a CMO complex. In 2023, brings us up to the present day. We have another, just getting more complex products being made by continuous manufacturing. This one, a peptide product. And then you also see the publication of ICH Q13, which is, again, a much broader scope than that original FDA guidance, because it covers not only drug products but also drug substances, and it's not limited by type of modality. And we'll discuss this more later in the presentation. So as we went through that continuous manufacturing journey, CDER had basically 3 types of programs that really supported us getting to where we are today in that journey. So one is focused on assessment inspection. That's the emerging technology program. But we also had key, in the science area. So investing in advanced manufacturing science and research was really important. And of course, taking those learnings both from assessment and from the research, and really helping us develop policy. And this led to the publication of ICHQ 13, which is actually a harmonized guidance for sponsors looking to implement continuous manufacturing. So I'll talk, for the rest of the presentation, talk a little about each of these different programs and how it relates to where we are in our continuous manufacturing journey. So the first program I'll talk to you about is the Emerging Technology Program. So this program has been around since 2014, as I mentioned. So many of you in the audience may be very familiar with this program, but for those who have not interacted with this program before, it's really, provides, it's really focused on promoting and facilitating adoption of innovative approaches to pharmaceutical product design and manufacturing. It's a cross-functional team. So today we have about 30 members, and it can always pull in additional ad hoc subject matter experts, and they represent all the relevant FDA quality review and inspection programs, including the Office of Pharmaceutical Quality, but also the Office of Compliance and the Office of Regulatory Affairs, really providing that one quality voice. And the program works by providing an opportunity for industry to engage and collaborate early with the FDA, discuss, identify, resolve technical and regulatory issues. And this early engagement can even before a specific product is identified. If you're considering an emerging technology that could be used across many different products, you can still approach the emerging technology program to discuss some of your early technical and regulatory questions. So the objectives of the program really kind of first of all serves as that centralized location for external inquiries from stakeholders, and really provides that forum for early engagement and dialogue, again, even if a particular product hasn't been yet identified. And from that early engagement, the program is designed to stick with that program throughout its maturing into eventually application, hopefully approval, and commercial production of a product. And that helps ensure the consistency and continuity and predictability in the review and inspection of that. And again, through that early engagement and all the way throughout that life cycle is identify and evaluate roadblocks, right? Whether related to existing policy or practice. And then as technologies mature, really to facilitate that knowledge transfer and help establish scientific standards and policy as needed. The ETP Emerging Technology Program is really focused on a collaborative approach. That means internally, in the FDA, collaborating, forming those cross-collaboration teams, as well as our engagement with industry. So it starts with our early engagement. Another unique aspect of the program is the emerging technology site visits. So these are non-inspection visits, but it gives us a chance to go out to the manufacturing facility where the continuous manufacturing or other innovative technology is being implemented and have more detailed discussions. And I can also engage in discussions focused on the pharmaceutical quality system, on readiness for inspection, and those kind of questions. Make sure the eventual application is as accessible as possible. But it continues on all the way through that when the application arrives, that integrated quality assessment. The ETP is serving as a co-lead of that application assessment and can participate in the pre-approval inspection, along with investigators from ORA. And the ETP has had many engagements related around continuous manufacturing. In fact, it's probably the technology we've had the most engagement with through the program. And through those engagements, we've kind of identified some of these control strategy considerations. So, one of that is that now you have material flowing through the process, and so as we may need to reevaluate some of the raw material specifications, right? Because there's new requirements for that material. The process is dynamic, and so we need to characterize those process dynamics. Typically it's done by characterizing the residence time distribution and understanding how the system's going to respond to if it's disturbed during the process. The process monitoring control strategy often involves process analytical technology, so that's usually a key discussion point. Increasingly, we've seen models being used as part of the control strategy, again to help us monitor and ensure product quality. Material collection and diversion. Because product is not being collected all at the end of the process, it's being collected over time. If there is an issue during the process, that material can be collected and diverted from the good product. There are known instances when this might be happen, such as startup and shutdown, but there could be unforeseen issues that might trigger this, and so there needs to be strategies to identify, isolate, and divert those materials if those do happen. And also there's the potential for real-time release testing, and we see many implementations of that for continuous manufacturing. So not only are there control strategy considerations, but there's considerations for the facility and quality management system. So there might be needed updates to the quality and production procedures. Often continuous manufacturing involves more automation, more collection of more process data, more process analytical technology, and so there needs to be sorting out of quality's oversight of those elements. Again, the equipment training is integrated, so not only are you performing your qualifications for individual pieces of equipment, but you need to focus on the qualification of the entire train. And if that equipment is being used for multiple products, there's things like cleaning and maintenance that needs to be, procedures for those need to be developed. Process validation. So, we see a lot of questions around this because a lot of the data developed during development, because it's being used to use the same process for continuous manufacturing, can actually give you knowledge, that can be used to support, process validation. So there's kind of a blurring a little bit of the individual steps for the process validation, but at the end of the day, you still need to demonstrate a robust process monitoring and control strategy. And again, if you do implement material diversion, what is the quality's role going to be in investigating those events? That needs to be kind of determined ahead of time. Advanced manufacturing research. So it's really important, as we talked about all those elements where the Emerging Technology Program has interacted with industry. It's really important the feedback on those points really be informed by the state-of-art manufacturing science, and that's really the goal of our research program. And there's really a dialogue with the Emerging Technology Program where questions can be raised to industry. They're looking for ETT feedback, and when the answers to those questions are, maybe not clear, or we need additional data, that's an opportunity for research to kind of support enhancing our understanding of that particular issue. And that research can be conducted, intramurally within the FDA, or it can be conducted through a collaboration, with academia or an outside partner. But really, the end goal is really to have shared learning and communications, both from the industry side, the FDA side, as those technologies get developed and are being implemented. And for advanced manufacturing, there's a number of different research areas our research program is focused on, including novel manufacturing operations. Sometimes switching to continuous doesn't mean just connecting pieces of equipment together. There might actually be new manufacturing operations. Again, we've looked at advanced manufacturing for complex products, so that brings an additional complexity to that, as well as ways to monitor control our processes, whether that's process analytical technology or process modeling. And the goal for these programs or these research projects is to really share the information that's generated from them. So over 100 publications have been produced from these projects to contribute to the community's knowledge. But that's not the only impact the research has had, because it has really directly informed our regulatory feedback and decision making. So It's either providing recommendations to the emerging technology program, really informing that, or informing the actual assessment of regulatory submissions that included continuous manufacturing. In fact, most of the submissions that have been approved by the FDA have been impacted by the FDA's research program. And also inform policy development, particularly in areas where we may not have a lot of regulatory experience yet. And that can be including elements of ICH Q13 or ICH Q5A, which is the guideline for viral safety. And when you're talking about continuous manufacturing for biotechnology products, that is a key potential issue. CDER, just to call out one example, CDER has done a lot of research on process models, and building those models has been actually applied to aid the assessment of a number of continuous manufacturing applications, supporting approval of those applications. So that work has also been recognized externally. Finally, I want to talk about, from the policy perspective, ICH Q13, which is a guideline on continuous manufacturing of drug substance, drug products. So, you can see there's a challenge when developing, ICH Q13, giving it broad scope, right? Is how to address all these considerations for, different types of processes, different types of modalities. And really, to tackle that, they came up with a strategy where they develop a main body of the guideline that covers fundamental aspects of continuous manufacturing that are not going to be specific to the technology or dosage form. So these contain some basic CM concepts, some scientific approaches, and regulatory considerations. Then they developed annexes that can provide illustrated examples and provide considerations specific maybe to a certain modality or production method. So in the CM concepts, it talks about, defining batch definition, because unlike batch manufacturing, continuous manufacturing, batch size is not necessarily constrained by the equipment size. And there can be flexible approaches, based on the quantity of output material or quantity of input material or runtime at a defined mass can be used to define the batch. And there's different modes of continuous manufacturing, right? We know that we've defined it as 2 or more integrated operations, but that can really be a partial, part of a process, or it can be the full, equipment train for a drug substance, drug product process, or it can even bridge, that drug substance, drug product process. For scientific approaches, it really focuses on this holistic approach to understanding process dynamics and designing the control strategy. Highlights the potential to use material diversion as part of the control strategy, which is unique to continuous manufacturing. It talks about the scientific considerations for changes in production output, whether you're changing the runtime or the mass flow rate, or scaling up the equipment. Each of those has different scientific considerations. It also identifies the potential of using the data generated during the process to really support continuous process verification as an ongoing way to verify the performance and the product quality. It also then talks about some regulatory considerations, clarifying where in the application certain continuous manufacturing specific information should be included, describing aspects of the pharmaceutical quality system with respect to managing disturbances and material diversion, highlights options to generate batches for primary stability data. It can be a single manufacturing run versus multiple shorter runs. There's different ways to kind of generate that key data. So finally, I would just like to conclude my talk with some concluding statements. So, when we look at the totality of these 3 programs, from the emerging technology program, research and policy, what has the impact of that been? And so we did a self-audit that looked at approved continuous manufacturing applications for solid oral drug products, which is where the most of our experience has been to date. And what we found is that actually, these applications actually had shorter times to approval and faster times to getting to the market than a cohort during a similar time period of batch manufacturing applications. For these particular products, that translated into an estimated between $100 and $500 million in early revenue for those companies. So a substantial financial benefit at helping to get market a little bit earlier. We looked at other aspects like, were there more post-approval changes for the continuous manufacturing processes, or was this impact on pre-approval inspections? We found actually for both of those, there was no really substantial differences between those two types of applications, whether we're talking about continuous manufacturing or batch manufacturing. Yeah, and looking at all these actions together, how did they address the regulatory hurdles that we heard, from industry? So one, early on in this journey, we heard, feedback from industry. We don't think we can implement continuous manufacturing without more engagement from the FDA, right? So, that led to, in part, the creation of the Emerging Technology Program, right, which is a program really focused on that early engagement and collaboration. You don't want to be, the first product approval of CM, right? You want to be that fast second. We hear a lot of that from industry, and then we saw in 2015, that first approved product manufacturing with continuous manufacturing was a big milestone. And since then, we've accepted over 50 proposals from industry concerning, concerning implementing continuous manufacturing into the Emerging Technology Program, and we've seen an additional approval of 15 products that use continuous manufacturing to make those products. So a big switch. Another feedback was, well, we were hesitant to implement until we have guidance from the FDA. So that has been, FDA has published draft guidance actually in 2019, and for the biotech side, published in 2022 about viral safety, which has for continuous biotech products, that's a key element for developing those types of processes. Another hurdle is like, well, we don't want to implement continuous manufacturing because we fear it's going to impact our approval timeline, right? I think the self-audit I just talked about showed that actually CM applications were approved faster than similar batch manufacturing, addressing that concern. Another concern is like, well, it's great, FDA is doing all this work, but what about, we produce for the globe and we have to interact with all these other regulators. So, we don't want to, implement CM without international, harmonization. And, this year, actually, the publication of the harmonized guidance of ICHQ 13 really is a milestone in addressing that concern. So I think, we continue to listen to, stakeholders as they are looking to implement these novel technologies that can have, impact for both their company as well as for patients, and trying to be responsive, to that feedback with our programs. And just some of the lessons learned as we went through this journey is that, again, that, we start off this journey with the idea that there could be potential benefits. And I see in a number of cases we've demonstrated, our industry has demonstrated that there are real benefits, maybe not for every product, doesn't make sense, but, a number of these instances, this implementation of continuous manufacturing really delivered actual benefits. Early engagement and collaboration is essential to get to where we are today. And, that's for folks, for us, who's been through the Emerging Technology Program, but really that early engagement collaboration was very key to making the progress we've seen to date. Science needs to lead the way. I think that investment, I talked about that journey starting in 2014, but it's actually not starting then, right? Because a lot of investment in the science and investment in the equipment happened actually before then, and that really was really key to enabling the success we've seen today, and the importance of FDA's Advanced Manufacturing Research Program to support our assessment and policy activities. Finally, it's important to engage, internationally. Pharmaceutical manufacturing is a global endeavor, so it's really important that we don't take that we really engage internationally. And so, I think that's been ICHQ 13 has been really important for that, and it provides a platform to build on, because that guidance has a number of annexes today, but you can see as new modalities get experience with continuous manufacturing, there could be new annexes added to that guidance over time to really grow the impact of that guideline. So that I'd just like to conclude by thanking some of my colleagues who contributed a lot to the material presented today. And finally, you just say thank you for your time and attention, and I look forward to the panel discussion later in the workshop. Thank you.
Hello everyone, my name is Tony Costa, and today I'll be presenting on continuous manufacturing platform for lipid and polymer-based nanoparticle therapeutics. Before I begin, I would like to thank FDA CDER, Small Business and Industry Assistance, for hosting this great event and giving me the opportunity to speak about some of our work on continuous manufacturing. At UConn, we received multiple awards from the FDA, spanning from around 2014 to 2022, and this work was all focused on continuous manufacturing. So back in 2014, this was really more of a proof of concept, understanding some of the basic material attributes, and performing a risk analysis on the various types of nanoparticles, mostly liposomes at the time, and really understanding how to make these nanoparticles using various methods. We selected a method, a solvent injection method, and then we expanded, looked at downstream processing, how to integrate different levels of PAT into the process stream, and then eventually in around the 2021, we actually developed a skid, and this industrial skid was actually brought to URI and used in a clean room setting to evaluate a liposomal doxorubicin formulation. Pharma industry, since inception, has relied on batch processing, and it has been a rather slow process to implement continuous manufacturing, which understandably is due to the strict requirements on drug products and reducing overall risks of product approvals. The 21st Century Cures Act was one initiative that opened the doors for advanced manufacturing technologies. At the FDA, OPQ has been leading the development of international guidelines, where these guidance for industry documents serve as a backbone for establishing frameworks and developing these continuous manufacturing processes. As a high-level overview, batch processing is carried out using multiple unit operations and requires multiple stages of testing and material storage. This type of process is volume dependent and may have difficulty in scaling, whereas a continuous process operates as long as raw material is fed into the system, with finished product being collected on the output. This type of process is time dependent and easier to scale. A continuous process has many potential benefits, for example, providing higher quality drug products, much more easier to develop controlled process streams, a faster to market approach, shaving off potentially months, and supporting end-to-end manufacturing, where we can combine upstream and downstream processes to have a state-of-the-art vaccine or drug product manufacturing platform. Here we show a liposomal nanoparticle, and the liposomal nanoparticle is made up of a lipid bilayer, which typically consists of phosphatidylcholine or other similar type of lipid, and these lipids may be saturated, unsaturated. They can have different hydrocarbon tail lengths. We also bring in cholesterol, which impacts the fluidity of the bilayer, and then some type of either charged lipid or pegylated lipid on the surface. The charged lipid and the pegylated lipid add to the stability of these nanoparticles in solution, or as a colloidal dispersion. Internal aqueous compartment is where an API, for example, a water-soluble API such as doxorubicin, may be encapsulated, and doxorubicin, when in the presence with certain salts, can form a linear crystal, and other API may actually form or load into the lipid bilayer, and also or a combination of the internal aqueous compartment and the lipid bilayer. The typical size range for these particles is around 40 nanometers on the low end, and then can really go beyond 500 nanometers to 1 micron. But for pharmaceutical applications, we're typically around the 100 nanometer diameter mark. Lipid nanoparticles and liposomes are both colloidal dispersions that share similar particle size characteristics and chemical makeup. LNPs include an ionizable lipid that directly interacts with nucleic acid and at certain ratios form a stabilized dispersion. Hybrid LNP liposome particles exist under certain conditions that form two compartments, a conventional aqueous core and a lipid nucleic acid matrix, potentially allowing for nucleic acid to exist in both compartments. A variety of nanoparticles that we produce using our nanoparticle processing system. These include liposomes, LNPs, and polymeric micelles. The bottom images show liposomes loaded with two different API, namely docetaxel and doxorubicin. On the bottom left, docetaxel liposomes form a unique, multifaceted structure. On the bottom right, liposomal doxorubicin forms a spherical particle with the linear doxorubicin crystal, whereas the combination of both API lead to an elongated, non-spherical particle. This nanoparticle processing system can be used as a tool to generate particles of various morphologies under a continuous operation. Now, from the onset of this work, we took a quality by design approach, which is really a risk-based approach to solving any problem. And on the left-hand side, we worked on multiple Ishikawa diagrams, and what these were really used for was to relate material attributes and process parameters to the potential critical quality attributes that we identified. We used guidance for industries to assist in pointing out some of the key factors or areas that we should be more focused on. But ultimately, this allowed us to make multiple lists, and these lists include material attributes and process parameters. For example, on the material attributes, lipid concentration, lipid pK, head group, ionizable lipid. On the process parameter side, we looked at different flow rates, temperatures, degrees of mixing, and then we looked to look at the functional relationships between the material attributes, process parameters, and how this all impacted the potential quality attributes. On this slide, we have a high-level process flowchart that outlines stages throughout the process stream. Starting on the left, we have an ethanol phase, which contains the pre-dissolved lipid and possibly a lipophilic API. There is also an aqueous 1 phase, which may be a buffer or a combination of a buffer plus water-soluble API, such as a nucleic acid or other water-soluble molecule. Nanoparticles are formed via a turbulent jet and then pass into a conditioning module and dilution port. At the dilution port, the material is diluted by an aqueous 2 phase that may be a different buffer than a 1 phase and used to adjust pH and salt concentrations. At the dilution port, we also have a particle size analyzer that measures online particle size. This is very useful as operators can understand in real-time conditions the particle size characteristics of their formulation. Downstream from the dilution port is a multi-stage TFF unit operating in single-pass mode, allowing for solvent reduction and buffer exchange. Another PA tool, designed as a soft sensor, is used to determine lipid concentration coming off the TFF segment. The material then passes to an active loading module, where certain API can be loaded into or onto the nanoparticle. A UV-vis spectrometer, also operating as a soft sensor, has been used to determine percent encapsulation in total API. Lastly, a purification stage, or a third TFF, is used to clean up the formulation, potentially reaching 99% encapsulation and providing a bioburden reduction stage. The material is then collected in a 2D bag, ready for further analysis and to proceed to fill finish. In this slide, we show videos of the nanoparticle formation site, or also the turbulent jet, at various flow rates. In each of the 4 sets of videos, the left video has a dye in the ethanol stream, whereas in the right video, we did not include any dye. The 30-second clip we observe here is actually taking place in about 1 second, and the 4 flow rates, ranging from 70 milliliters per minute to 300 milliliters per minute, all show conditions where a stabilized jet is formed. Typically, at the lower 70 milliliter per minute flow rate, larger nanoparticles are formed, and at the higher 300 milliliter per minute flow rate, smaller nanoparticles are formed. In a continuous manufacturing process, Process Analytical Technology is a required tool that provides data in real time and can be used to control and/or monitor a process. The PAT requirements are important to identify as they can impact other controls in the process stream and data collection rates. PAT can be installed inline, online, or at-line. Online and inline allow for material to continue downstream post-measurement, whereas at-line measurements require material sampling and subsequent discarding of the material. Some of the PAT tools are developed as soft sensors with predictive algorithms. Having these tools allows operators and feedback control mechanisms to make decisions and meet specifications. Dynamic light scattering to determine particle size, turbidity measurements to relate to nanoparticle concentrations, and UV-Vis spectroscopy to measure drug encapsulation and total API are just some of the sensors that can be used as tools to control a continuous process stream, ultimately providing a robust nanoparticle platform. One of the setups that we've used was a nanoflow sizer. This is a spatially resolved dynamic light scattering system. It's by in-process LSP. It allows you to attach a flow cell onto the process, and you can actually flow at rather high flow rates and very accurately measure particle size. We've been able to measure individual data points in less than 6 seconds. So this really leads to a rapid online analysis. You can see in real time what particles are being formed. Does it meet your specification for particle size, and is it acceptable within a certain concentration range? So it really gives you a much better picture of what you're developing, and not having to wait till the end of a process stream to see if you are actually within your specification. Example, particle size is being monitored in real time. The z-average particle size, which is one measure of the diameter of the particle along with the Pauli dispersity index, are being measured. Other attributes such as shear rate and temperature are also recorded and analyzed. From the particle size graph, the size is very consistent around 92 to 93 nanometers, and the PDI is averaging around 0.05, indicating a rather monodispersed particle population. Having inline-online PAT in a process stream enables scientists and engineers to relate intermediate nanoparticles to the end product, which can lead to a better understanding of the particle behavior throughout all the downstream processing. Here's an example of the online doxorubicin and lipid concentration analysis. So what we did here is we added doxorubicin for the top graph, added doxorubicin, and then you can see the doxorubicin went up to about 95% encapsulation. So soon after the dox was added, we instantly see a 95% encapsulation reading. Then we stopped, continued to run the system after removal of the doxorubicin, and added again at a higher temperature down at a later time point, and then we were even able to get above 95%, almost closer to 96, 97% encapsulation. So that was just simply by adjusting temperatures at the loading site. With the continuous process, we're able to load doxorubicin within seconds, as opposed to a batch process that may actually take multiple hours to heat up a large container and to load all at once, whereas this continuous process is loading small amounts over a period of time and loading a smaller number of nanoparticles over a period of time, but we're able to do so in a very short period of time. Lipid concentration analysis, we're looking at what's coming off the TFF, so we know that we're within the specification, and for this we were targeting around 25 millimolar lipid, and we were definitely within reason of the specification throughout the duration of the run. Now I just want to go into a design of experiment example. So what we did here is we looked at really mostly material attributes and some process parameters. So we looked at post-formation temperature, aqueous phase flow rate, cholesterol percentage, main lipid type, and the aqueous phase salt. So PBS, phosphate buffered saline, phosphate buffer, or sodium chloride. And both the pH values for the PBS and the phosphate buffer were set to 7.45. And then as a response, we're looking at the particle size, polydispersity index, and zeta potential. These HSPC-based liposomes, a predictive model was generated for particle size and an R-squared of 0.89 was obtained for the 270 runs. We compared the datasets using multiple surface plots and also grouped the datasets. One example is shown in the graphs on the left, which is the particle size data plotted against particle size formation, temperature, and flow rate. These examples are grouped based on 30% or 50% cholesterol. As can be seen from these surface plots, it is that the blue surface profile, which represents phosphate buffered saline, has always the largest particles. The red, which is phosphate buffer, and the gray, sodium chloride, surface profiles are similar for their particle size data. Now, we can use this data to help in-process optimization and a control strategy. And you may notice there are other areas of the surface profiles that are somewhat flat, meaning the particle size does not change with temperature or flow rate. These zones can be used to generate particles with a wider tolerance on process parameters. With that said, these types of design of experiments and surface profiles provide a tremendous amount of data, and the continuous processing system we developed can rapidly produce particles for these studies in a short period of time. Design of experiment study, more of an extended study. We included DOPC as part of the main lipid type. DOPC has unsaturated hydrocarbon tails and a low phase transition temperature compared to other lipids. Across all flow rates and temperatures, the particle size for DOPC liposomes was less than 50 nanometers in diameter, which are very small liposomal particles. DMPC, which has 14 carbons on its saturated hydrocarbon chains, DPPC, which has 16 carbons on its saturated hydrocarbon chain, and DSPC, which has 18 carbons on its saturated hydrocarbon chains, all formed much larger particles. DMPC was able to form the largest particles up to about 350 nanometers in diameter, whereas DSPC formed the smallest particles for the saturated hydrocarbon lipids. In the case of DSPC, only smaller particles formed due to its high phase transition and more solid-like bilayer. Accordingly, small formulation adjustments in the number of carbons on a hydrocarbon chain or bond saturation can greatly impact bilayer physical characteristics and the nanoparticle structure, all of which must be fully considered in pharmaceutical development work. The continuous processing system that we developed is also used to generate polymeric micelles consisting of a block copolymer of MPEG BPCl. Curcumin was used as a hydrophobic model drug, and TEM was used to analyze the particle morphology and structure. We can see consistent particle size and no worm-like or elongated structures for these particles in the top left images. Polarized light microscopy, which are the images on the right, was used as a technique to determine the entrapped model drug, as crystallized curcumin is evident when only in water and not present upon loading in the polymeric micelles, which is A versus B. In addition, the bottom left image of the blank micelles, which is a clear solution, the loaded micelles, which is a clear yellow solution, versus the free curcumin, which is a very yellow opaque solution, all indicates full loading into this nanoparticle system. We continued this work with paclitaxel, and the graph on the bottom right shows an in vitro dissolution with first order release for the micellized paclitaxel, and also as compared to Genaxol PM, which is a commercial product. We have a case study on liposomal doxorubicin. This work was performed at URI's Pharmaceutical Development Institute Training Center. We successfully transported the nanoparticle system from Yukon to URI for this work. Here is the specification for liposomal doxorubicin. Our focus was to match the parameters in the specification to our final formulation while using a continuous process. In this manner, we sought to meet the lipid composition, the final lipid concentration, the total amount of doxorubicin, the loading battery, final external buffer, and the particle size. Now, looking at the system that we set up in the clean room area, and again, this is our first system that we developed here. So it was brought into a clean room. We did some initial validation on the system, cleanliness, swabs, internal, external, surface testing, the air quality testing in the room. So we went through multiple procedures to understand what we would need for SOPs and just general protocols. And then in addition, we were set up to run for liposomal doxorubicin. So looking at the procedures and overviews that we were able to develop over this time, so we looked at gowning procedures, draft batch records, draft cleaning SOPs. For the cleaning, we loaded the system as a cleaning validation. We loaded the system with the sucrose, and then we removed, and we looked at the total TOC endotoxin levels, different system SOPs that we provided. Also then environmental monitoring and cleaning cycle development, and then also personnel monitoring for the process. On the right-hand side, we show a picture of a general recipe. So this is built into the software on the system, and this gives you really full control over all of the process streams. And for example, changing flow rates and running multiple steps. And this gives you, full control over this process. Particle size, so we're definitely in the range that we anticipated. So we had these 3 runs that were performed. Actually, 2 runs were on one day, another run was on a different day. So we were able to form these particles, 87.3 nanometer in diameter. Very nice, very low PDI, 0.03. Encapsulation a little bit lower than what we were expecting based on our previous runs, around 86%. However, these were run a work in progress. The heating stage was set to a lower value than what we probably needed to run at, so that would have easily brought the temperature up. But we were limited in certain circumstances for this particular run while the system was at this facility. So in that case, we did get a little bit lower, but 86% is still a very nice encapsulation for a single pass encapsulation. So overall, runs were very successful, and these low standard deviations and percent errors were reported. EM images on this slide demonstrate a clear picture. Continuous processing has the ability to produce more uniform particles when compared to batch processing. The technology developed at UConn on the left have majority spherical and linear doxorubicin crystal structures. The brand product in the center have more elongated and less spherical particles. It can also be seen empty liposomes and possibly free drug and needle-like structures. The generic product on the right, which has a wide range of particle sizes and morphologies, with some large empty particles and needle-like structures present. Inherently, the loading processes are similar, but the time scale changes significantly when a continuous process is used. Being able to load the API in a matter of seconds versus tens of minutes to hours can lead to a more efficient active loading process. Overall, a continuous process provides the tunability required to optimize a process stream. In the continuous process, we can feed this back into models to better understand and predict the process. For example, as shown in the plots on the left, we can determine concentration steps and dilution steps required to meet salt and total drug concentrations. Accordingly, these process parameters can then be fed into a recipe as an optimized condition. On the right, a subset of the overall data is displayed for a typical run, indicating the enormous amount of information we can generate from a continuous process. Next, having the ability to really control the crystal morphology. So this is an interesting aspect that we were not planning when we started off with these with this work. We were looking at liposomal doxorubicin, and most of the time we were making nice linear crystals. But then we started on some formulations to see that we can actually form majority of the crystals as a circular crystal. And on the right hand side, you can clearly see all these circular crystals that are present, and even the ones that actually are that appear to be linear. If we used a cryo-TM tilt at 45 degree, and we're able to actually see that these are not linear crystals, these are actually spherical crystals. When we tilted the images on the left, we could see that these were actually linear crystals and not, they also were not spherical. There's a couple spherical crystals in there, however, you can see one or two present in this image. So that may be another difference between continuous processing, is that you're able to go from linear structure to circular structure of an API within this type of microenvironment of a liposome. So very interesting, how could this potentially impact anti-cancer therapies, and is there a benefit of having a linear versus circular crystal on any potential dissolution or release mechanism? And then, in general, understanding the structures of the liposomal doxorubicin. So you can, and see here the elongated linear nanocrystal on the left. We have more of a spherical linear nanocrystal. So it looks like the crystal size is roughly the same with respect to the number of fibers within the crystal. But then when you look at a spherical circular nanocrystal, you can see how it wraps around and how it forms this very nice circular type of morphology. And then you have some potential U-shaped, so these are going to be circular, or over a longer period of time they connect to form circular. So these are just caught as an intermediate type of nanocrystal. And then all this comes down, how does this impact crystal? How does this crystal structure impact potential clinical adverse events, PK data, etc. Whereas liposomal doxorubicin is to determine the individual particle aspect ratio. This is done by using software such as ImageJ to compare the shortest and longest axes, where a ratio of 1 indicates a perfect sphere, and an aspect ratio greater than 1 indicates the elongated particle, for example, especially above a value of 1.2. From the graph on this slide, the formulation that was developed using a continuous process, which is in red, had all the particles with an aspect ratio of less than 1.15, indicating very spherical populations, whereas Doxil had many particles above an aspect ratio of 1.25 and even out to about 2.0. This indicates much more elongated particles present in that population. And now we also looked at different orthogonal sizing techniques, so cryo-TEM, number distribution, and actually just basically counting the particles and looking at their particle size using an ImageJ approach. Or we have a microfluidic resistive pulse sensing. This is a Spectrodyne NCS1. We tested the particles, and we can, and this is more of a volume distribution. And then looking at the cumulant, DLS intensity-based analysis, and all these, in all three approaches, we were able to see very similar particle size trends. If you get a very low PDI, 0.029, so really anything less than 0.05, we get more uniform particle populations. As we get above 0.05, which most people talk about 0.1 being, an indication of monodispersity, but as you get above even 0.05, you start to see potentially that there may be some larger particles or smaller particles, mixed in with the formulation, which is picked up, more predominantly by a cryo-TM or this volume distribution technique. And again, then going back down to larger particles, 124 nanometers in size, low PDI on DLS, and we can also see, both, respective the volume and number distribution techniques. Now, another question is, when looking at these nanoparticles, how it's being measured, by what technique, and even within the same technique, for example, dynamic light scattering, you could have potentially two different light sources So 633 nanometers, that could be a laser source versus just a standard light source. And so looking at a light source of 1300 nanometers, you really do get different results. So this has to go into a control strategy, understanding what system was used, what light source was used, and what are the expectations. In one case here we see that we get higher PDI using a 1300 nanometer light source. In both these cases actually we see higher PDI, and then in this other case we see we get a very, very low PDI, extremely monodispersed, 0.008 and 0.039. So, which one is acceptable? Should we accept 0.1 to be acceptable for nanoparticles, 0.2, or 0.05? So really, this helps point us in the right direction of different strategies that we can develop. As a liposomal doxorubicin in vivo study, we were selected by the Nanotechnology Characterization Laboratory at Frederick National Laboratory for Cancer Research, and this was a great opportunity for the university where we were able to work together and provide them some of our nanoparticles and review the results that they were able to generate with their in vivo study. For this study, we looked at two different formulations, namely NCL425, that was designed to have a linear doxorubicin crystal, and NCL426, that was designed to have a circular doxorubicin crystal. Using cryo-TEM, the number, particle size, aspect ratio, and percent linear crystals, we were able to analyze these formulations. Formulation 426 had over 86% linear crystals, while formulation 425 had over 50% of the circular crystals. However, most likely this is much greater, as a tilt was not done on the cryo-TEM, and as we've seen previously, this would have led to a much greater number of circular crystals. Overall, the aspect ratio of both of these formulations were close to 1, which indicates a near spherical particle. So interestingly, when we look at the z-average particle size, and this is what we can use for inline analysis, we're getting exactly what we were picking up around the 87 nanometer mark for the z-average, and very low PDI. So that's a very nice indication that I was using different dilution strategies, 100x versus 1000x, and we don't really see major differences between the two, which is a good indication that dilution is not impacting the overall particle size which is expected for this type of nanoparticle. And then for the NCL26, we see the same. We're getting around 120 nanometer particles, and then also very, very nice low PDI. Formulation stability studies were performed. An example data set from NCL426 is shown here. AF4 separation with inline mols and DLS was performed on these samples, and the shape factor shift was analyzed for 426 with and without plasma. The minimal shape factor shift observed indicates minimal protein binding, consistent with the commercial drug products. And we saw the same results for the NCL425 as well. A toxicity evaluation was performed for the two formulations in comparison to a marketed product. The levels of doxorubicin and doxorubicinol in the heart, liver, and ear tissue were analyzed. Both formulations had comparable values to the manufactured product. Statistically significant differences were found in doxorubicinol concentrations in the heart and liver, indicating slight variations in tissue concentration profiles. The objective of the study was to evaluate potential bioequivalence of NCL425 and NCL426 in comparison to the commercial product Doxil. PK data for the total, unencapsulated doxorubicin, unbound doxorubicin, and doxorubicinol were analyzed. As shown in the above graph, all these profiles were similar and most likely would have similar biologic effect. However, bioequivalence was not fully met. The purpose of the study was not to necessarily meet a bioequivalence, but to understand potential differences due to linear versus circular doxorubicin crystal formulations. we look at an mRNA LNP case study, where we mostly looked at particle morphology and how that relates to some of the process conditions. So some of the mRNA LNPs that we formed, what we'd like to look at here was how do the different process parameters and processing strategies impact these types of nanoparticles. And one case here is really focusing on the intermediates. So combination of including or leaving these particles in ethanol and also using a low phase transition lipid led to more of a delamination or blob-like appearance on the outer layer. So you can see as it appears that there's this outer bilayer that's coming off or that's associated with the particles. Then when we have low phase transition lipid and we leave them in a high percent of ethanol for a period of time, we can really see the particles swell, they get much larger, and then they start to delaminate, and there's multiple compartments that are provided. However, when you use a higher phase transition lipid, you reduce the amount of ethanol present, you can get much more of a nice solid core, no separation, very stable structure for these LNPs. So all of this can really be helpful in developing and optimizing your nanoparticle formulation. For the mRNA LNP transfection, we looked at just a 28-day study, and we could see that we were able to in a cell line for K562 chronic myelogenous leukemia cells, we were able to detect a response, and depending on the high versus low concentration of the mRNA, and clearly significant compared to just a control PBS and also the naked mRNA. Now, another area that we focused on some of these grants was on the CFD, the computational fluid dynamics, and also on the MD. And we have a very nice paper that was published in Molecular Pharmaceutics that explains the coarse-grained molecular dynamic simulations of paclitaxel-loaded polymeric micelles. So on the CFD aspect, we looked at the different jet flows, and we did simulations, really that adds information that is more difficult to actually measure. We looked at an eddy current analysis, which includes just what's called the delta criterion, and what this is looking at more is the vortices rotation that takes place within these jet streams, and we're trying to relate more of an eddy current or vortice that's being formed with some of the material attributes and some of the resulting quality attributes. So, for example, particle size. On the MD side, we wanted to understand more of the basic mechanism of the particle formation. And in this case, starting with just individual lipid molecules and water molecules and ethanol molecules, and then going over multiple nanoseconds, so 10 nanoseconds, 100 nanoseconds, 500 nanoseconds, all the way to 1800 nanoseconds to understand really how the particles may form. And in this potential mechanism, we can see the individual particles coming together, and then they form more of a linear plate, and then this linear plate structure then bends inward, and then starts to form a sphere, and it closes up. So this is a nice indication of how potentially the API becomes encapsulated, and also then leading us to understand the general formation process. Lastly, for this work, we looked at shear simulations, so understanding how when mixing, potentially with high degrees of mixing versus a low degree of mixing, and we can see that the shear before shear and after shear particle size was able to be decreased. So this may have some useful impacts, and where we can relate MD to CFD as a new means to really understand a process going from atomic scale all the way up to to more of a bulk, to bulk analysis. Now on the paclitaxel side, we looked at different PEG, PLA, micelle formation, and for paclitaxel, what was interesting here with this work is we're able to see how the paclitaxel enters the micelle and general the time frame really taking place similar to a liposome around the 1800 to 2000 nanoseconds. In this slide, we see the polymeric micelle structures that can actually form. So on the left-hand side, we have a blank spherical structure. Then we have a drug-loaded spherical structure, and then a drug-loaded rod-like structure, and a drug-loaded worm-like structure. So all of these structures can easily form when producing these particles. So even though this may be a thermodynamically stable system, the actual physical mixing process can also impact and have a high degree of influence on these types of structures. We did find that under a higher shear, higher mixing environment, we would form these spherical structures, and then and lower polydispersity index, and then under lower mixing, low shear conditions, we could actually form more of a worm-like structure, and this would be indicated by a higher polydispersity sample on the experimental side. And another aspect of this work is we're able to look at the free energy of the polymeric micelle dissociation. So again, something that's rather maybe difficult to measure, but we can use this MD approach to understand the different potentials along for say pulling a single PEG PLA and pulling a single paclitaxel molecule away from the nanoparticle, and you can see their degrees of the free energy. So even all the way up to about 80-90 kilocalories per mole versus around 40 kilocalories per mole for paclitaxel. So this indicating drug release versus particle dissociation. And lastly, we worked on artificial neural networks for continuous manufacturing. We took in really an abundance of data that we collected over the years. We added in some of the MD, CFD aspects, and we started to look at this as different models. So multiple input, multiple output model, and also multiple input, single output model. So MIMO versus MISO for predicting the liposome particle size and PDI. And then we looked at the different training sets and testing stages. And this is where we're able to actually determine, give you a general idea of liposomal particle size. So if you wanted to say, I'm going to increase my aqueous flow rate and my ethanol flow rate and my osmolarity, and I'm going to be switching, cholesterol from 20% to 60%, and also my hydrocarbon tail length, this at what temperature, this will give you an idea of the size of the particle that you may generate. So really, again, great tool, understanding and giving you the ability to really generate any particle size. Thank you everyone for your attention, and now I would like to briefly acknowledge all the people that were part of this work. Firstly, the US FDA for providing us with these funding opportunities and for all their support over the years. At UConn, we had a large team of graduate students and postdocs over the years, and this work helped train these students and postdocs for new careers in the exciting and burgeoning nanoparticle field. The PDI Training Center at URI for their support in the operating of our system in a cleanroom setting, and the UMass Chan Med School for the outstanding cryo-TM images. Lastly, to NCL for all their work on the liposomal doxorubicin characterization and in vivo studies.
Thank you for the great presentations. We'll now enter our break until 10:45 a.m. Eastern Time. Please enjoy your break. Welcome back from the break. We hope you're able to refresh your coffee or tea to help move us right into the remaining presentation before the Q&A panel. Our next presentation will cover an integrated platform for continuous RNA nanoparticle formulation and drying by Dr. Kurt Ristroff from Purdue University. Please join me to welcome Dr. Ristroff.
All right, everybody. My name is Kurt Ristroff. I'm an assistant professor of agricultural and biological engineering at Purdue University. I'm very happy to talk to you today about our work on an integrated platform for continuous RNA nanoparticle formulation and drying. This is a grant that's funded by the FDA BAA mechanism. And we're working in collaboration with Professor Robert Prudhomme at Princeton University and Dr. Mark Castenton and his team at Saran Bioscience, which is a contract research and manufacturing organization in Oregon. So, to begin with, I'm going to be talking about the nanoparticle formulation process that we use called flash nanoprecipitation, how it works, and how we use it in particular for preparing lipid nanoparticles to encapsulate mRNA. Then, in the second part, I'll be talking about specific instances where we've used FNP for drug delivery in general, nano-encapsulation, nano-formulation, a bunch of different types of drugs from different chemical classes. Yes, I know today is mostly about continuous manufacturing for lipid nanoparticles, but we're going to talk about some work that led up to our current work on lipid nanoparticles, which is going to be described in part 1. Okay, so some potential benefits of nanoparticle encapsulation are shown on this slide. So just starting and going counterclockwise from the left, one of the main benefits that you want is to improve the circulation time in the blood or the residence time in the body of a particular active ingredient that you're trying to encapsulate. So many of the biological molecules of interest in the pharmaceutical world today are what we call biopharmaceuticals, biologics. These are peptides, proteins, and nucleic acids. The body can easily recognize these as foreign agents and really quickly clear them from the blood if they're injected, for example. So in order to shield them from those processes of the body, to allow those drugs to have more time to do their work, we want to encapsulate them into some nanoparticle carrier that is small enough that it can still move around in the body, but provides a surface layer that is potentially invisible to the immune system, for example. Next on the list is targeting. So one of the things that people are trying is to coat the outside of those nanoparticle delivery vehicles with some chemistry that will allow it to target to specific tissues, specific organs, or specific cell types. For example, macrophages, other white blood cells, or tumors. The next bullet point here is co-delivery of multiple payloads. So let's say that you have two different drugs from different chemical classes that aren't very physically compatible with one another, right? You can imagine, well, many drugs are given as cocktails where you have multiple drugs that are supposed to act together in concert to improve what we call therapeutic synergism. Okay, if the drugs are very physically different from one another, they may not be easy to dose at the same time, right? And if you want those drugs to reach the same place in the body at the same time, we and others have shown, okay, if you co-encapsulate these things into the same nanoparticle, into the same drug delivery vehicle, then you can physically package them together and get them, optimize their chances of reaching their target tissue at the same time. Over on the right-hand side of the slide, we talk about penetrating physiological mucus. This has applications for both oral drugs and drugs that are given as an injection or an inhalation, for example. Physiological mucus is a major physiological barrier. A lot of drugs get caught in that physiological mucus. That's what it's there for, is to catch foreign agents. And so, for example, you have an infection that's underneath the mucus layer. It's very hard to get a drug down underneath that mucus layer to treat those, let's say it's bacteria, for example. So if you can package your drug in a delivery vehicle that can penetrate through that mucus layer, then you could potentially reach the site of infection better. This is all about trying to get drugs where they need to go better. The next bullet point is controlled or stimulus responsive release. So for example, for a lot of these biologic drugs, biopharmaceuticals, peptides, proteins, and nucleic acids, maybe you want them to stay inside of their delivery vehicle until they reach the place where you want them to do their work. And maybe there's something different about the place that you want them to do their work. Maybe the pH is a little bit different. Maybe the temperature is a little bit different. So if you can design a drug delivery vehicle that is temperature responsive or pH responsive or responds to some enzyme that's present at your target location, then maybe you can induce drug release sort of on demand. Once the drug or once the delivery vehicle gets to where it's going, it can somehow sense where it is and know to let go its payload in that location. And then the last example here is for hydrophobic drugs, for things that don't dissolve in water, we use nanoencapsulation to improve their dissolution kinetics in the gastrointestinal tract. Okay, so the technique that we're bringing to the table is flash nanoprecipitation. This was developed in Professor Prudhomme's lab over the last 20 years or so as a strategy of encapsulating very strongly hydrophobic small molecule drugs. It was originally designed for small molecule cancer drugs, which needed delivery solutions, needed formulation and delivery solutions because their solubility in water was basically nil. So the way this strategy works, like other nanoprecipitation processes, which I'll talk a little bit about, is you can take a strongly hydrophobic drug, shown here on the, shown here on the, in the yellow dots here on the left-hand side of this slide. This drug won't dissolve in water, but it can dissolve in an organic solvent. So if you dissolve this along with a amphiphilic stabilizer, that's a molecule with some hydrophobic region and some hydrophilic region, all these things will dissolve in a organic solvent. Then if you rapidly mix that organic solvent against water, then the result will be a mixed solvent environment that has too much water for the hydrophobic things to stay dissolved. They will then start to precipitate. Okay, they will be diffusing around, and as hydrophobic things find one another by diffusion, they'll stick together. Okay, and this will form your nanoparticle core when your drugs stick together, and then the amphiphilic stabilizer will adhere to the surface of that growing particle core and prevent further growth. So that's shown as a timeline on the right-hand side of the graphs, or on the right-hand side of the slide. If you do your mixing at time 0, if you get a well-mixed system very quickly, then all of this self-assembly can happen homogeneously if your system is already well-mixed by the time all of this is happening. And you can end up with what we call core shell nanoparticles that have a core consisting of the drug you were trying to encapsulate and a shell consisting of, let's say, that amphiphilic polymer on the surface. And you see there are a couple of real tricks to this. One is that the process, the game is over after 50 to 100 milliseconds if you're making, let's say, a 100 nanometer particle. So self-assembly happens really, really quickly. All right, we're going to talk about why that's important. By the way, what you get out of this, like I said, it's a core shell nanoparticle. We know how to tune the size of the particle, and if we have a hydrophobic drug in the core, usually our assembly is so fast that the drug doesn't have time to crystallize, so we can trap it in an amorphous form, which is a benefit for us. Okay, if you don't do your mixing very fast, then over the time scale of that particle assembly, you can end up with non-homogeneous precipitation. Okay, that's undesirable. So you can see here I've used red to denote regions of the organic solvent, and blue to denote regions of the water, and purple to denote the mixed region. If your mixing is slower than your particle assembly, then you'll have some regions with lots of drug and lots of water, and some regions with lots of polymer and lots of organic solvent. You'll have this inhomogeneous, non-homogeneous solution, and therefore your particle assembly will also be heterogeneous, and you won't end up with as reproducible of a system as if your mixing is really, really fast. So in flash nanoprecipitation, our mixers are set up, and the geometries of our mixers are set up such that when we introduce these streams into the mixers under what we call turbulent flow, so this is fast mixing, we get homogeneity on the order of about 1.5 milliseconds, which is much faster than the 50 to 100 milliseconds that it takes for our particles to assemble. So that's one of the key takeaways for flash nanoprecipitation is that you want your mixing to be faster than your particle assembly. And particle assembly isn't something you can really control the time frame of. It's going to be that 60 to 100 milliseconds, and so therefore if you want to be in the game, you want your mixing to be really, really fast, much faster than that 50 to 60 milliseconds. We use turbulent mixing to achieve this. The other key point is that all of your compounds, your core and your stabilizer, have to precipitate on basically a similar time scale to one another. Okay? If your drug precipitates much more slowly than your polymer, then your polymer will self-assemble with itself and form these empty polymer micelles before the drug has a chance to precipitate. What that means in practice is that we need usually very hydrophobic drugs for this precipitation process. We're going to talk about an exception to that, a major exception to that, over the next couple of slides. So these are two of the main points. By the way, this is not a T mixer. This is, we call this a confined impinging jets mixer. There are two geometries for flash nanoprecipitation mixers. I'll show those in a couple of slides. So there's a cavity inside the mixer that allows that turbulent field to evolve and gives us the really good fast mixing. So it's not exactly like a T mixer like you'd get from a chromatography tubing. Okay. So for nanoparticles that we make by flash nanoprecipitation, we get a very dense layer of polymer on the surface, which is attractive for things like mucus diffusion. We can have mixtures of surface polymers as long as they're all properly amphiphilic, and we achieve independent core and shell chemistries. Okay, so if a core is hydrophobic enough to precipitate, we can trap it with a bunch of different polymer options and vice versa. Okay, and when your core material is sufficiently hydrophobic, if it has a log P, where P is the partition coefficient between octanol and water, greater than about 4.5, we're usually seeing about 100% encapsulation efficiency of that really strongly hydrophobic drug, and we can get particles that are up to, recently up to 90 weight% drug by mass, which is attractive from a cost of goods standpoint. If your delivery vehicle is mostly the drug that you're trying to deliver, then you're not paying a bunch of money for other things other than your drugs. Okay, so a couple of advantages from flash nanoprecipitation. The process is scalable. I'll talk about that more in a couple of slides. And it's very reproducible. Because your mixing is happening so fast and all of the majority of your particle assembly is happening under that well-mixed environment, we get very reproducible particle assembly, and the process can be run continuously. So one of these mixers here, for example, the only moving parts are the streams themselves. So as long as we have good pumps to pump the two fluid streams through the mixer, we can continuously operate. Challenges for F&P include, well, what if you have a drug that's not hydrophobic enough to precipitate really fast on the same time scales as your amphiphilic polymer? And if we're looking at things like global health applications, malaria or tuberculosis, or if you're looking at the agrochemical space, we need extremely low-cost excipients. Even if the only excipient we're using is the polymer stabilizer itself, we need to find very, very, very low-cost options so that the overall cost of goods of that formulation will be low. Okay, here are the two different mixing geometries that we have for flash nanoprecipitation. The first one here is called a confined impinging jets mixer, and you can see that cavity that allows the turbulent field to evolve. Here is a computational fluid dynamics calculation of where the point of mixing is the highest. So you see the point of mixing is highest right in the center of the mixer, and then lower on the outside of the mixer. This geometry requires that the two streams coming in be of almost equal flow rates so that all the mixing can happen in this cavity. In contrast, we have a 4-inlet mixer called the multi-inlet vortex mixer, we call it. This guy has 4 inlets, and particle assembly and mixing all happens in the center here. And again, you can see on the CFD graphs here that the intensity of the mixing is highest right in the center of the MIVM, which is really attractive because we want our assembly to all happen away from the walls. We don't want any of our hydrophobic materials to stick to the walls of our mixer. That could potentially lead to fouling. So here our mixing, our really good mixing and our precipitation are occurring in the center of our mixer, which is good. One of the benefits of this particular this particular 4-inlet mixer geometry is that our mixer, our streams don't have to have equal velocities like they do here. So here the two streams have to have equal flow rates, but here we have much more flexibility in those. Okay, I'm going to talk a little bit about the scalability of this continuous process now. Here are 3 different scale FNP mixers that we have in the lab. Here's the confined impinging jets mixer. It outputs 60 milliliters of particles a minute. Here's one of our MIVMs that outputs 1.5 liters a minute, and here's a larger scale MIVM that operates at outputs 5 liters of nanoparticles per minute. And for reference, the MIVM, the 5-liter MIVM here is about 5 or 6 inches in diameter. So this whole thing, it's still a fairly small device, and there's fundamentally no limitation on us designing a even larger scale mixer if we wanted to. Usually the limiting factors are getting a good pump and the cost of goods for the materials that we're encapsulating. Okay, I'm going to go through why the process is scalable and why it contrasts a little bit to the microfluidic mixing that some people do for nanoprecipitation. Nanoprecipitation occurs when you do this mixing between something that's dissolved in an organic solvent and water to drive precipitation of it. So we can either have mixing that uses turbulent flow, like flash nanoprecipitation. The other kind of flow is laminar flow, which is the type of flow used by microfluidic devices. So turbulence, for the engineers in the audience, you're probably familiar with this. For those of you who don't know about Reynolds number, this is the Reynolds number. It's a dimensionless group that is the product of the density of a fluid, the velocity of a fluid, and the channel size that it's flowing in, divided by the viscosity of the fluid. Okay, so if you want to mix 2 fluids, let's say you want to mix a fluid, this Reynolds number is a function of the velocity and the channel size. So if you want turbulent mixing, you have to have high Reynolds numbers. Okay, so Reynolds number, traditionally Reynolds numbers below about 8,000 are non-turbulent, and Reynolds numbers above about 8,000 are turbulent flow. Okay, so if you want turbulent flow, you have to have a high Reynolds number, which for a fixed fluid where you can't change the density or the viscosity means you have to have a high velocity or a high channel dimension. In contrast, for microfluidics, if you need a low Reynolds number, so the only knobs you can tune are velocity and channel size, you want those both to be low. Okay, so microfluidic mixing is very reproducible and there are many elegant, very beautiful science papers about using microfluidic mixing for a variety of things. We use turbulent mixing in part because of this next equation. Q here is the volumetric flow rate coming out of the mixer. So Q, like Reynolds number, is a function of the velocity and the diameter of your channels. So Q is pi v d squared over 4. So you can see that if you're using turbulent flow, you're in a Reynolds number regime where you need your velocities to be high and you need your channel sizes to be large. Okay, those two contribute straight away to a high volumetric throughput. Okay, what does that mean? That means that when you use turbulent mixing, when you use turbulent flow to drive your mixing, your process is inherently scalable. It's inherently large scale. By going up in Reynolds number, by going up in velocity and channel diameter, you're building in higher and higher throughput. And in contrast, by staying at those very low Reynolds numbers, the laminar flow mixing gives you an inherently lower throughput. Okay, so we say turbulent mixing is one, your mixing is fast, which is very attractive, and 2, your throughput is built in. Okay, and here's an example of this. You can see this is data from a collaboration that we had with a contract research organization who scaled up one of our formulations. We were making exactly the same nanoparticles in a 5 milliliter batch in the lab that they were making at the 3,000 liter scale in industry. Okay, this is a graph, by the way, this is a particle size distribution graph where we have the diameter of our particles on the x-axis and the intensity of our particle scattering on the y-axis. Okay, so you can use this flash nanoprecipitation rapid mixing setup to make lipid nanoparticles. If you want to encapsulate mRNA, for example, you load your mRNA in the anti-solvent water stream that you're using to induce precipitation of your hydrophobic lipids. So here these lipid nanoparticles are made of 4 different kinds of lipids. There is a zwitterionic phospholipid that helps to form the interface of the nanoparticle. There's cholesterol that helps the structure of the particle. There's a PEG lipid, that's a lipid that serves the role of the amphiphilic stabilizer from the previous examples. This sits at the surface and has a hydrophilic polymer on the shown here in purple. And then you have a cationizable lipid. This is a lipid that is cationic below a certain pH. And why we use this? Well, it's cationic, which we'll use to form an ion pair with the anionic phosphate groups on the backbone of your nucleic acid polymer. That's half of the molecule, that's the cationic half. The other half are these hydrophobic tails that add hydrophobicity to your anionic mRNA. So basically these two guys will come together and form an electrostatic interaction. It's like an ion pair. And the resulting ion paired complex will no longer be charged because all the charges have found a partner, an oppositely charged partner, and will have sufficient hydrophobicity to precipitate. And that hydrophobicity is coming from the hydrophobic tails on the cationizable lipid. So these guys come together and form a hydrophobic salt of mRNA that will precipitate along with your zwitterionic phospholipid, your cholesterol, and your PEG lipid. That forms your nanoparticle. And again, we want to do all of this under really fast mixing such that the electrostatic interaction and ion pairing between the cationizable lipid and the mRNA, and then the subsequent precipitation of that complex along with the precipitation of all the other hydrophobic lipids. Everything is too hydrophobic in this mixed solvent environment to remain dissolved, so it all precipitates out. We want all of that to be homogeneous, and so therefore we want our mixing to be as fast as possible. We want our mixing to be much faster than the time that it takes for all of that precipitation and ion pairing to occur, which is in the time scale of, again, 60 to 100 milliseconds or so. So here on the right-hand side of this slide are 4 different particle size distributions for different lipid nanoparticles that we've made encapsulating different RNAs. One's an mRNA, one's a small interfering RNA, one has 2 different kinds of mRNA co-loaded together, which is very important for some applications. And the last one has an mRNA co-loaded with some proteins. So let's say now that we've explained at length the flash nanoprecipitation process, but that's just one step in the overall procedure for manufacturing mRNA lipid nanoparticles. Okay, so nanoprecipitation comes first. I put flash nanoprecipitation, which is Prudhomme's way of making using turbulent flow to drive really fast mixing, but there are other forms of nanoprecipitation. You'll hear about another very good rapid mixing process from Dr. Costa later on today, and I think from some others over the course of today. So anyway, you can sub in your favorite mixing process for this block. Then what you have coming out of that mixing process is mRNA lipid nanoparticles that are in some mixture of usually water and ethanol. Okay, the ethanol was used to dissolve all the lipids in the previous slide, and then the water was used to dissolve the mRNA. So what do you do with your effluent stream that came out of your mixer? I've shown, well, one arrow is going into tangential flow filtration or diafiltration. This is another continuous process that's used for buffer exchange. So let's say, for example, that you form your lipid nanoparticles at a pH of about 5.5, which is required for making sure that those cationic lipids that you're using to ion pair the mRNA are properly charged. That's a good pH for nanoprecipitation, for driving that ion pairing complexation, but it may not be the best pH to store your lipid nanoparticles after they've been made. So if you want to change the pH, or you want to swap out the salts that you used during precipitation for better salts for storage, you can use a process like tangential flow filtration or diafiltration. Like I said, again, this is a continuous process. It's also a very standard pharmaceutical process that's been implemented at the large scale, so we know how to do this. You can also use this to remove any residual ethanol from your particles, which is desirable from a stability standpoint and from an injectability standpoint. Okay, but you notice there's an offshoot branch from our effluent mixer, our mixer effluent, that goes into what I'm calling the analytical box. Okay, so if you're continuously making your particles, you will want to sample from your effluent, from your mixer. We say F&P is very reproducible. There are other very reproducible large-scale mixing processes, but of course with any industrial process you want to have inline sensors to tell if what you're making is in spec or not. So here are some of the analytical techniques that are employed for lipid nanoparticles. The first and sort of easiest one is dynamic light scattering, which tells you what the size of your nanoparticles is. Okay, I've shown you a couple of particle size distribution graphs over the last several slides. That's that particle size on the x-axis, particle light scattering intensity on the y-axis graph. This is where that comes from, and you can install dynamic light scattering either in line, right downstream of your mixer and before you go into TFF, or you can pull an aliquot from your mixer effluent and go check by offline DLS. Okay, and there's, I think, significant discussion today and throughout the industry and in the regulatory space about where exactly you want these different analytical techniques to be in your overall process. Do you want them all to be in line? If so, how do you make them all in line, et cetera? Another assay that's used is the ribogreen assay. This measures mRNA encapsulation efficiency, which we expect to change as a function of things like pH and ionic strength. So when you're thinking about what buffer you make your particles in versus what buffer you diafilter your particles into, using the ribogreen assay will help you ensure that your mRNA is still encapsulated. And then another question is whether your mRNA has degraded or not. The stability of these formulations is a significant concern to a lot of different people. And so the technique there, you can use capillary or gel electrophoresis to tell if your mRNA has remained chemically stable or if it started to break down. After tangential flow filtration, where maybe you've put your particles in the right buffer, you've got a couple of options. So you can fill into vials and freeze right away, and that can be your final dosage form. This was the final dosage form for the Moderna and Pfizer COVID-19 lipid nanoparticle vaccines. Those were, remember, shipped around the world at low temperatures as a frozen block of ice. That's one option. You could also go from freezing into freeze drying. This is an option that a lot of people are exploring. This is also called lyophilization. This is when you use a low pressure chamber to drive sublimation of the ice in your frozen sample. Basically remove all the water in the form of ice from your sample, and it leaves you with a dry powder that you can then redisperse in water prior to injection. As an alternative to this freezing and freeze drying route, you could look at some other drying operations. And why are we looking at these drying and freezing operations? This is for storage. This is because most of these lipid nanoparticles are not going to be made and used right away. This is part of the manufacturing challenge. So let's say that you want to remove the water from your particles. There are a couple of reasons why you would want to do this. Stability, shipping, and storage, generally. So water is expensive to ship. It's heavy. We also think that water facilitates degradation of the mRNA that's encapsulated into your particles. So if you can dry these out, we expect that you'll get a much more stable powder over time, which is good. And maybe it doesn't even have to be stored at those very, very low temperatures. So that's why we're looking at other drying operations. Spray drying is one option there. Spray drying is a traditional process that's used in the food and pharmaceutical industries. We'll talk about that in a little bit. Spray freeze drying is in between freeze drying and spray drying. Then there's also an option like electrostatic spray drying, which is an alternative form of spray drying. Okay, and this will give you your final dosage form. You can take your final dosage form then and continue to do these analytical tests. If your final dosage form is a powder, you would redisperse it prior to doing some of these assessments. If your final dosage form is a frozen vial, then you would thaw it out prior to doing these, and you would use this to check stability over time. Looking at some of the other options a little bit in more detail, there are pros and cons to these different downstream processing operations that you want to think about. This is part of what we're doing in the grant, is we're trying to compare these different downstream options to one another, using as our upstream way of forming nanoparticles the flash nanoprecipitation procedure. Okay, so spray drying is continuous, it's large scale, it has medium energy intensity, it's not the most energetically expensive process there is, and it gives you a flowable powder, which is desirable for a couple of different dosage forms, like a dry powder inhaler, right? Not all mRNA lipid nanoparticles are expected to be given as an intramuscular injection in somebody's arm. That works fine for vaccines, but if you want to have, for example, something that's delivered by dry powder inhaler, you'd want a more flowable powder. Okay. Cons of spray drying: it is a process that uses elevated temperatures, and there's concern around degrading the mRNA. Spray drying is also something that's, practically speaking, it's very difficult to optimize at the lab scale because it requires a lot of material, and at very low batch sizes, you're not getting a lot of material out of the process that you can characterize. So actually, spray drying is one of those processes that's hard to scale down, and the minimum sort of lab scale that people work at, it requires a lot of lipid nanoparticles to run, and lipids are expensive, and mRNA is expensive, so that it's a little bit tricky to optimize, particularly at the academic lab scale, I'll say. Spray freeze drying works at lower temperatures, which is good for the mRNA stability, and it gives you a flowable powder still, but it requires very high energy intensity, and it's a batch-wise process. Lyophilization, or freeze drying, is fairly similar. It works at very low temperatures. It's a really well-established unit operation, but it has some cons noted here. It's a batch-wise process. It has high energy intensity. The lyophilized cake that you get out isn't a very flowable powder. It's not as easy to load into something like a dry powder inhaler, and this lyophilization process does require that you freeze your particles prior to freeze drying, which is its own unit operation that brings its own challenges. Okay, so this is some of what we're doing in this grant, right, is we're looking at the overall process of lipid nanoparticle manufacture. We say, well, we've got the very good upstream unit operation figured out. We know this flash nanoprecipitation works really well. It gives you this really good mixing. It gives you very homogeneous particle assembly. It gives you really reproducible production, and it can and has been scaled up in the pharmaceutical industry. So now we want to compare several of these different downstream unit operations to try and find some of the best ones, and we're working on potentially improving some of those unit operations, optimizing them for lipid nanoparticle manufacturing. Okay, so that's the first part. In the second part, I'm just going to go quickly through a couple of examples of how we've used flash nanoprecipitation to formulate small molecules in biopharmaceuticals. And the point of these examples is to show you a couple of those downstream unit operations in action. So for example, we wanted to work with some drugs for global health indications, so malaria, tuberculosis, etc. So we had to adapt the FNP process, which I showed you on slide 2 or 3, to work with very, very low-cost excipients. So, for example, we swapped out the diblock copolymer stabilizer that we traditionally use in our polymer nanoparticles, which are different from lipid nanoparticles only in that there's a diblock copolymer on the surface and that the core is made up of mostly our drug rather than a mixture of lipids. Anyway, we swapped out the diblock copolymer stabilizer for another amphiphilic polymer that was significantly cheaper. This is HPMCAS is a common pharmaceutical enteric polymer that's used in tableting all the time. It turns out that that can stabilize our nanoparticles against aggregation, which is really attractive, and its cost is attractive from the global health standpoint. The other thing, another adaptation to FNP that we've done, I've sort of already shown you this, but incorporating the idea of hydrophobic ion pairing. So this is how you make the lipid nanoparticles, is you take something that's hydrophilic and won't precipitate in a traditional nanoprecipitation process like this, and you make basically a hydrophobic salt out of it by ion pairing it with an oppositely charged hydrophobic molecule that can be loaded into your organic stream. And then you can add a diblock copolymer stabilizer, for example, and get your polymer nanoparticle that's almost entirely loaded with drug, which is attractive. This works really nicely for small molecules that need to be ion paired to drive precipitation, or smaller things like peptides or proteins. For RNA, you have to go all the way to a lipid nanoparticle because you need this particular mixture of lipids for good particle formation for biological activity. Okay, so here's an example of improving the bioavailability of a strongly hydrophobic drug. This is lumefantrin. It's an anti-malarial. It's extremely hydrophobic, and we wanted to make a solid oral dosage form that could be given to, say, a mother in Southeast Africa, or Sub-Saharan Africa, or Southeast Asia rather, and is given in the form of a paper sachet that can be opened and mixed with water and then fed to an infant, for example. So we couldn't make a tablet, we couldn't make a capsule, we wanted to make a powder that would improve the dissolution kinetics of this drug, because, like I said, it's very strongly hydrophobic, so if you take it by mouth, it won't dissolve in the time that you have, in the time that it's in your GI tract. So we took lumefantrin and put it through our flash nanoprecipitation process using this HPMCAS low-cost polymer as the stabilizer. We got 200 nanometer particles that had 90 weight % lumefantrin by mass. We then put those through tangential flow filtration to concentrate them, and then put them through spray drying. So some of these unit operations are familiar to you now that we've talked about what downstream of nanoprecipitation may look like. So here is a graph of a particle size distribution graph of the lumefantrin nanoparticles before and after all these different unit operations, and what you'll see is these curves are all basically the same, where the particle size was the same right after flash nanoprecipitation, after TFF, and then the powder that you get out of spray drying, if you add that back to water, we see that our nanoparticles redisperse back to pretty much their original size, which is really attractive. Okay, and then when you feed these to mice, or I think rats in this case, in an animal test, we saw that the bioavailability of the lumefantrin was improved 4.2-fold compared to the unprocessed drug, and that bioavailability held up after the powder was aged at 75% relative humidity and 50 degrees Celsius in a vial for a week. So this is a graph of plasma concentration of lumefantrin in the blood of the rats over time, and you see that the black and the orange, which are our nanoparticle powders, outpace the unprocessed lumefantrin quite significantly. Note that this is a logarithmic axis. Okay. As another example, we can, pivoting gears a little bit, we're going to move to flash nanoprecipitation with hydrophobic ion pairing for encapsulating. Here we're going to talk about polymyxin B, which is a peptide antibiotic that's commonly used. We wanted to encapsulate this using this hydrophobic ion pairing approach using flash nanoprecipitation, and demonstrate not only that we could encapsulate it, but then also find whether there was any improved efficacy of the drug from being encapsulated into a nanoparticle, like what I talked about on the first slide. These benefits of nanoencapsulation. Are we getting controlled release? Are we getting better circulation time, etc.? So what we found was we could encapsulate polymyxin B, whose structure is shown here. It has these 5 positively charged groups along its sequence. We ion paired this with a very common fatty acid found primarily in olive oil, or is the primary component in olive oil rather. This is oleic acid. These will form a hydrophobic ion pair, and that hydrophobic ion pair will precipitate and can be stabilized by a diblock copolymer. We found that by changing the ratio of oleic acid to polymyxin, we could tune the release rate of polymyxin from the nanoparticles when we incubated them in a salt solution, which was really exciting. So you see we get the fastest release when we have one equivalent of oleate, and then slower and slower release as we're adding more and more of this hydrophobic counterion, which translated to really nice improved efficacy in a mouse lung infection model that we did with Jian Li at Monash. So here we're these mice have a bacterial infection, and we're delivering either unencapsulated polymyxin B or our nanoparticles through an intratracheal route, and finding that this is the log 10 of colony forming units per lung. We find that our nanoparticles significantly reduce the number of bacteria that are in the lung over time. So this is no treatment, this is treatment with free drug, and then this is treatment with our nanoparticles. Okay. Another quick example, we can use this hydrophobic ion pairing flash nanoprecipitation combined approach to encapsulate proteins also. So here we encapsulated lysozyme, which is an enzyme with a molecular weight of about 14.4 kilodaltons. We got basically 100% encapsulation efficiency, and the exciting part here was that this lysozyme is an enzyme, and we showed that its activity was maintained after it was encapsulated and released. So making a hydrophobic ion pair out of it and precipitating it into these nanoparticles didn't affect its enzymatic activity, which was really exciting to see. Over here on the right side, we see another protein we encapsulated, ovalbumin. This is a good model antigen for making vaccines. So we got 90% encapsulation efficiency, and showed that when we dosed these nanoparticles nasally into a mouse model, we got 10-fold improvement in immunogenicity of our nanoparticles compared to free ovalbumin. And again, we think this is because we're seeing controlled release over time, and we're seeing things like the ability of our particles to diffuse through mucus, so they can reach the immune system more easily to be recognized and induce that immune response that we were going after. Last example real quick, we put, when you encapsulate molecules into these nanoparticles, you can also take advantage of the surface chemistry that you install. So here we put a targeting moiety on the end of the polymer chains that we were using to stabilize our nanoparticles. Here that we were encapsulating an anti-tubercular peptide. Tuberculosis lives inside of macrophages, so we put a mannose moiety on the end of these polymers to go after the mannose receptor that's on the surface of macrophages, thinking that, well, if the macrophage wants to uptake mannose, if we put mannose on the surface of our particles, then the macrophage will uptake the particles more, and that means that our anti-tubercular peptide can get at the tuberculosis that's inside the macrophages. And this is what we found. If you had particles that had no mannose on the surface, they were indistinguishable from the control. As you start to if 4% of the polymer chains on the surface of your particle have mannose on the end, we get, what is that, a log and a half knockdown of our tuberculosis, and if 70% of the polymer chains on the surface of our particle have that group, we get another 3/4 of a log unit of knockdown, which was really good to see. So this is an example of a targeting story that we can do with our flash nanoprecipitation produced nanoparticles. Okay. Here's a snapshot of my lab just to close. We're looking at co-encapsulate. Well, we're very interested in all those sorts of things I've been talking about so far, and we're really excited to continue working with the FDA. We're very interested in the idea of co-encapsulating these biologics along with hydrophobic small molecules. I talked about this a little bit in the beginning with combination therapy. We're looking at how, when you do this hydrophobic ion pairing, to make hydrophobic salts out of peptides, proteins, or nucleic acids, those hydrophobic salts pack into different liquid crystalline phases in the cores of your nanoparticles, which is interesting and is the mechanism behind the different release rates that we see. So we're studying that. We're looking at unit operations for making nanoparticles at scale, like we're talking about. We have this very good flash nanoprecipitation platform for making nanoparticles, and then we want to look at things like TFF, spray drying, and other downstream ways of supporting that. And then also, I mentioned that we are working with very low cost nanoparticle formulations for example, global health indications, and also in the agrochemical space. We're trying to do nano delivery of many different biological actives for plants. I'm currently looking to hire some postdocs and graduate students. If anybody knows any good candidates, please do encourage them to contact me. None of this would have been possible without the very hardworking members, both of this overall grant team, Professor Prudhomme, Mark Castleton and his team at Saran, and then also my team here at Purdue and our different funding agencies and partners across industry and academia. Lastly, I'll put in a plug for the Department of Agricultural and Biological Engineering at Purdue. Both our undergrad and grad programs have been ranked number 1 or 2 in the U.S. for 13 years. We have a new building. Both the College of Agriculture and College of Engineering at Purdue are very, very highly regarded. And we sit in both, actually. We're a unique department that sits in both colleges. So lots of opportunities for undergraduate and graduate study here. Thanks very much, everybody. I'm looking forward to hearing your questions during the panel discussion and talking more about lipid nanoparticle manufacture, scale up, and processing. Thanks very much.
Thank you all for the wonderful presentations. We'll now enter into our first Q&A panel. As a reminder, if you haven't entered your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. The first group of questions will be directed to Dr. Xiaoming Xu, and here is the first question for Dr. Xu. In continuous manufacturing, how do you determine the batch record and batch size? Dr. Xu, we're not able to hear you at this time. What we're going to do is we're going to let our tech team reach out to you. We're going to move on to our next presenter, Dr. Thomas O'Connor, and then we'll check back in with you a little bit later on. So here is the question for Dr. O'Connor. If we can use CM for early phase clinical trial materials, what is the expectation for process validation?
Yeah, thank you, Ray, for the good question there. Yeah, there's the expectations are for process validation, are the same as if you were using a batch process, especially if you're in the early phase, clinical trial stages. We don't expect the process to be validated at that stage. You would have phase appropriate information. I will say that, the aspect of a lot of continuous operations, because of the intensified processes, there's often opportunity maybe not have scale up or, later down development, which means a lot of the data you generate during your clinical manufacturing runs may be able to be leveraged to support process validation as you get ready for commercial manufacturing. So that is an opportunity that continues can potentially provide. So thank you for that question.
Thank you for responding to that question. We've got a couple more for Dr. O'Connor, and here is the next question. How do we make proposals to be considered in ETP? Do we request this during a pre-IND meeting, or is there a separate pathway?
Yeah, this is a great question. So thank you for that question. So we can do there's 2 ways I think you can do this. So and it's spelled out in the emergent technology guidance, and you can send a proposal to the program to be admitted to the program. And so the first thing we would need to know is a little bit about the technology you're proposing and why you consider it novel. And the positive impact you expect to have on product quality. And then once accepted into the emergent technology program, if you already have a molecule identified that you're using it with, you already have a development program going on, you can submit that pre-IND or an IND amendment with your questions to the agency, and just make sure you highlight that you've been accepted in the emergent technology program so that we can be involved and support addressing those questions. If you don't have a molecule yet identified and you're kind of using this maybe as a platform, looking across different potential drug candidates, but you have questions about that you could help your development program, you can submit a meeting package to the ETT directly without having an IND in-house yet, and to get that early feedback. So that's also an opportunity the program provides. So thank you.
Thank you for responding to that question. One final question for Dr. O'Connor is the following. Why do you think the early CM applications are approved faster?
Yes, thank you for that question. There's two aspects that we found. So we found the approval times are faster, and as well as the marketing time. So I'll address the approval first. So it is, I think, important to note that those early drug candidates that use the continuous manufacturing were also a number of them were breakthrough drug candidates, had that kind of clinical impact. So I mean that drove some of it, but I also think the early engagement through the ATT was a significant factor as well, because through that mechanism there was a lot of alignment already pre-built before the application arrives. So a lot of that early engagement really sets the stage for a very I would say smooth application assessment process. And then we saw the acceleration to market. So even after approval, getting that market quicker. And I think that's related to the technology. And this idea of this potential opportunity in the scale up and simplifying tech transfer that we saw that impact there. So thank you.
Thank you for responding to that group of questions. We're going to check back in with Dr. Xiaoming Su to see if his mic is adjusted. And here's the question. We'll read that again. In continuous manufacturing, how do you determine the batch record and batch size?
Hopefully the second try works. Thanks for the question. So as Tom mentioned in his talk, that a lot of definition that's provided in the ICH Q7, it's also relevant for continuous manufacturing, especially in terms of the batch size. Generally, this either is defined by the quantity of the material, whether it's input material or the output, or can be defined based on the runtime at a fixed mass flow rate. So I would highly recommend refer to the ICH guidelines, so specifically Q7 and also Q13, as Tom mentioned. Q13 is a guidance more specific to the continuous manufacturing of drug substance and drug product. So thank you.
Thank you, Dr. Xu. We hear you loud and clear. We have a few more questions for you, and here's the next question. Switching from batch to continuous manufacturing may be great and easy for brand companies. Is there ever a case also to be made for generic companies?
Thank you. This is a great question. As we've heard from Tom's talk, that there's a lot of perceived barriers, potentially with any disruptive technologies like continuous manufacturing. So as we are seeing already with the solid oral, some of the successful examples of approved continuous manufacturing drug product, there is actually quite a benefit, and the actual perceived barrier may not be that high. For example, as Tom mentioned, time to market and also early engagement that's possible with the FDA, which allowed facilitate the development of the product. That being said, we do recognize that developing a generic version of drug product is oftentimes different than innovator product. The margin of kind of operation is more rather thin. So I think the key would be to identify the right product to develop and to start the conversion from batch to continuous. So in my opinion, so one category of the product that potentially can be really a good kind of opportunity is complex formulations. So examples include what we heard today, liposomes, lipid nanoparticles, and also maybe things like formulation like long-acting injectables like polymicromicelles, PLGA microspheres. So all of those products, when the manufacturing process are often more complex and multiple steps, where the quality sometimes is hard to kind of build into the product, and the failure rate when if it's high. So this is could be an ideal candidate or the product, potentially category product to be switching from batch to continuous. And also I think from the generic standpoint, oftentimes you're looking at transferring knowledge from one product to the other product, which is an ideal example to actually utilize something like continuous, because it allows you to adopt the knowledge that you have learned from developing one continuous process and apply it to the next. So overall, I think there's a lot of potential, even in the development of the generic space, to utilizing continuous manufacturing. So thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Xu, and here's the next question. For continuous manufacturing of lipid nanoparticles, what are the areas that still need more research?
Yeah, thank you for the question. So we all probably should celebrate what we have accomplished already in the lipid nanoparticle space, especially with the success of the mRNA COVID vaccine, as we heard earlier today. But I think there are still areas that we can improve. One possible area is in the material characterization, whether it's the lipid or whether it's the cargo, which is oligonucleotide or RNAs. What are the analytical techniques that is suitable to provide us more information and better information to allow us to better control and screen the material and utilize in the production? The other, I think, is an area, it's the analytical technology that may be particularly suited for process monitoring and process controls. So this means like process analytical technologies. So this potentially can enable what we consider as real-time release, RTRT, release testing. So this does offer flexibility, but it requires analytical process that's more suitable, sensitive, and robust to the manufacturing process. And also the other area I think could be potentially useful and to explore further is in the modeling and simulation space. As you heard from Tony's talk earlier about use of CFD and MD simulations, which potentially can speed up some of the understanding process about the manufacturing process or the formation process, which in the end, in turn, may benefit the optimization of the manufacturing process, maybe implementing better control strategies. So I do think there are still areas that we can still research on, and I think that's a lot of what at UConn, at Purdue, and at Princeton, where FDA has funded multiple research, is really trying to generating some more knowledge in that space. So thank you.
Thank you for responding to that question. We do have one more question for Dr. Xu, and here's the question. What will be the challenge to scale up the manufacturing of lipid nanoparticles?
Thank you for the question. I think actually this question, I think Kurt has already talked about in his talk about the flash nanoprecipitation, about the challenges and the benefit of utilizing the continuous processes to overcome some of the scale up issues or challenges. And also I think from the other angle to look at it, if we looking at the continuous processes, and one of the critical benefit is that the product throughput is oftentimes defined or dictated by the runtime if the process is operated at a steady state conditions. In that case, the quantity of the material output is really dependent on the throughput and the runtime. mass flow rate and the runtime. So in a way, you may have less of a challenge or issue with the need to scale up the manufacturing if the desire is to generating larger quantity of material. Instead, you could have run the process longer to produce enough material. Be considered as a batch, but I will maybe keep this as a question also for Kurt to comment on later on when he's maybe able to chime in. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in for Antonio Costa, and here's the first question for Dr. Costa. How well understood is the docetaxel doxorubicin co-loading leading to the elongated particles?
Yes, thank you, and hello everyone. So I would say, with respect to the docetaxel, what we see here is it does have a log P, so it's more of around 4.2, so it is more of a hydrophobic molecule. We can see from the cryo-TEM that there's a multifaceted structure with respect to the nanoparticle. So what this can lead to is more of a lipid domains, boundary effects within within the bilayer itself. There could be potential phase separations as well. And then when this potentially lead to more of a flexible bilayer. So when we combine the doxorubicin and we have the internal environment that allows for the ammonium sulfate high salt concentration, which allows for the doxorubicin to form a crystal. This could be the cause which would lead to more of an elongated particle. So it's almost as we add the docetaxel into the particle or into the mix. This is added actually from the ethanol phase, whereas the doxorubicin is added downstream after the particles are already formed. So we're actually loading and adding at two different locations in the continuous process. So this could also potentially impact the particle formation, but most likely, it's the relationship between the hydrophobicity of the docetaxel, the interaction with the lipid, which allows for more of an elongated structure to form once the crystal, the doxorubicin crystal, forms on the inside. Thank you.
Thank you for responding to that question. And the next question for Dr. Costa is the following. Why do you measure shear rate?
Yeah, so the shear rate that's measured is actually more for the nanoparticle, the nanoflow sizer, which is the inline particle analysis unit that we used. So the shear is actually more important to understand at the boundary where the particles are actually being measured for their particle size, that they're within acceptable limits, and that's more specific to that particular instrument. So it has to be if it's at a certain range. If you're above that range, then you have other convective effects that would be influencing the particle size measurement. So we just have to always make sure that we're within that certain range.
Thank you for responding to that question. We've got a couple more questions for Dr. Costa, and here's the next question. Can you explain how to measure turbidity and what is the range of acceptance?
Yeah, so for turbidity, it's an NIR approach that was used, but it's really it's more or less looking at the sample with respect to at certain concentrations. So we're able to measure based on concentration units, which is a more of an absorbance measurement, and then there's also scattering effects. So this is measured typically in a ppm. And you're looking at more of the effects of scattering on the particles, and then the combination of these two, we can understand, we can relate to the overall nanoparticle concentration and turbidity of the sample. So this is important. When developing models, we can use this for predictive models. We can understand relationships between other characteristics of the formulation and relate this overall to the concentration. So acceptable range would really depend on the sensor, would depend on the sensor of choice, and depend on the particles that you're measuring. So really small particles will give you much lower values, and larger particles would really increase the turbidity measurement. So ultimately understanding really the sensor and your sample is important. So there's not really a specific range that would be acceptable. Thank you.
Thank you for responding to that question. We have one more question for Dr. Costa, and here's the question. What DOE software was used for the modeling?
Yeah, so we used the JMP by SAS, and with that software, really gives you a lot of flexibility in the different types of models that we can use, full factorial, partial factorial designs, and other complex designs, or for example, d-optimal design. So this allows you to generate the models, run the experiment, and after you run the experiments, input all the data. So really, it's a easy to use, great software. There's plenty of other softwares out there that can, that can be used as well. But that's the one that we use for this work. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in for Dr. Kurt Rystroff, and here is the first question for Dr. Rystroff. Can you use flash nanoprecipitation for hydrophilic API, and what is the success rate?
Thanks very much for that question. So this hydrophobic ion pairing strategy that I talked about toward the back half of the presentation, we can use that for several classes of things that you may or may not call hydrophilic. We can use that for weakly hydrophobic small molecules or biologics that have a log P maybe in that 2 to 3 range, which aren't hydrophobic enough to precipitate by flash nanoprecipitation, but if we increase their hydrophobicity using that ion pairing approach, we can. Then we've extended the FNP plus hydrophobic ion pairing platform to very soluble biologics. Polymyxin B has a log P of -5 or -6 or something like that. So to date, we've encapsulated a couple dozen strongly hydrophilic peptides and proteins using that combined approach. And we're usually, I think the lowest encapsulation efficiency we've found with something that actually formed good particles is something like 88 or 90%. So it's high encapsulation efficiency. And what I mean by things that make good particles, if you have let's say a molecule like a dextran, it's a water-soluble compound, but it doesn't have any ionizable groups that we can take advantage of in order to ion pair and encapsulate. So if you don't have ionizable residues, you can't use that approach.
Thank you for responding to that question. I've got a few more questions that came in for Dr. Rystroff, and here's the next question. Is flash nanoprecipitation limited to producing spherical core shell solid nanoparticles, or is there flexibility to produce other shapes or other products, for example, liposomes?
That's a great question. Thank you. We haven't made liposomes with FNP because of the rate of precipitation. What we get out are these solid lipid nanoparticles or core shell polymer particles. Also, because of the chemistry of self-assembly, we almost exclusively get spherical particles, yes. So if you want something with a very high aspect ratio, that's harder to achieve. There has been some work by Rod Priestley in chemical engineering at Princeton on using that rapid mixing process to make Janus particles that have these are spherical particles that have 2 faces that are composed of two different polymers that phase separate. They're both hydrophobic, so they will co-precipitate into a single particle, but then they will spontaneously phase separate. So you get something that has two hemispheres that are made of different material. But those still have an aspect ratio of 1. They're still spherical. But so yeah, generally we don't get anything other than spherical particles out of this. Thank you.
Thank you for responding to that question. The next question for Dr. Rystroff is the following. What is the role of block copolymer stabilizer? Is it not part of the final nanoparticles?
Yep, the block copolymer adsorbs onto the surface of the growing nanoparticle core and prevents further aggregation. So you get that first nucleation step where the core starts to assemble. And if you didn't have any diblock copolymer stabilizer or stabilizer, there would be nothing to prevent the hydrophobic core material from continuing to aggregate and forming into a big crystal. So we can stabilize these particles either with a diblock copolymer or with what the LNPs are stabilized with is a PEG lipid, usually a DMG PEG that has a fairly small lipid group and then about a 2 kilodalton PEG group. You can also use some other smaller surfactant-like molecules. Lecithin, soybean lecithin is a zwitterionic phospholipid that can be used as the surface stabilizer. I showed HPMCAS is also amphiphilic enough to be the surface stabilizer. So that's where it ends up, is on the surface. Thanks.
Thank you for responding to that question. This will be the final question for Dr. Rystroff in this round. And here's the question. You showed that the LMN formulation showed the same size distribution after spraying, drying, and redispersion in MQ water. I assume the powder would be finally redispersed in tap water for final application. Do you have size distribution data of such a suspension as well, and what would this look like?
Well, that's a great question. So the question's about the lumefantrine formulations. You know, when they're actually applied in sub-Saharan Africa or Southeast Asia, are they going to be mixed with milieu water? No, they'll be mixed with whatever water's available. So we don't really expect the composition of the water, as long as the water's, well, we were told by the Gates Foundation to assume that water would be potable, but small differences in ionic strength or pH shouldn't affect the redispersibility of those powders into the final dosage form. We have not tested that, but actually that's a very good idea, and I've got some contacts in Nigeria who we could potentially reach out to and try and get water to use for that. It's a good idea. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist. We've got a few more questions that came in from Dr. Xiaoming Xu. And here is the next question for Dr. Xu. Depo-Dur is an extended release liposome formulation of morphine sulfate. Are there any special specific or regulatory considerations if applying continuous manufacturing techniques to produce this type of liposomes or similar liposomes containing a controlled substance?
Thank you for the question. So what you were referring to, the Depo-Dur, is one of the multivesicular liposomes that's produced through emulsification process. So generally the size is much larger than what we consider typical liposomes. The multivesicular liposomes generally are in the micron size range. So we have not seen the commercial, at least application of the continuous manufacturing in producing this type of multivesicular liposomes. However, we do believe there is a potential of utilizing the continuous processes, such as the one that you heard today, to generating this type of dispersion. Essentially, it's oil in water kind of dispersion colloidal system to start with when you're producing the multivesicular liposome. Instead of oil, of course, this is the organic solvent we're talking about. So there is potential, and in general, I think in terms of the feasibility, when the processes involve liquid mixing, I think there is always potential of using the continuous process to produce the particles. Now, I think still some research work is needed in order to see the commercial success. But as I mentioned in my earlier beginning introduction to the symposium, I think sometimes we need to think bigger. We need to think forward about what we hope to utilize. I think if you think about the potential benefit of the utilized continuous process to produce multiple multivesicular liposome, is that you may be able to produce more homogeneous the type of particles, which we know has been a challenge. And also you may be able to better control the internal morphology, which is a characteristic kind of feature of the multivesicular liposome. So using continuous process, we believe there are some potential benefit to further improve the consistency of the manufacturing. And internally, actually, we're working on this category product, looking at how do we kind of switching from batch processing to continuous processing for something like emulsification process. Hopefully there are more interest and there are more development in the academia and in the industry on this topic in general. But thank you.
Thank you for responding to that question. The next question for Dr. Xu is the following. What is the proper step to test size PDI zeta to be filled later for registration purposes, for example, after making the nanoparticles only, or after purification only, or after concentration, or at the stage of the processing?
So thank you for the question. So my understanding of the question perhaps is related to what's where or when it's appropriate to perform a particle size measurement on the polydispersity index, or is it a potential in order to control the product? I think the answer to that is certainly you need at the end of the kind of product manufacturing before releasing it. So this is what typically considered as a batch release test. And so you would need to set the acceptance criteria or specification for particle size or zeta potential or other quality attribute. And then you would need to develop method to properly measure and control that attribute. And however, sometimes, especially we know with some of the more complex processes. And also maybe another benefit of the continuous manufacturing is that you're actually able to measure those quality attributes in line during the production through the measurement like process analytical technology or PAT, which allow you to see the quality of the product that you're producing in real time. So you may be able to start measure intermediate quality attributes such as after liposome or lipid nanoparticle formation, or after loading, or after purification. So those I think will be critical to measure in order to build confidence for the downstream process. So because again, it's a continuous operation, so all the units are linked together. So one deviation in one process may impact the downstream process. So the ability to measure in real time those quality attributes will really give you insight into how the process, how the product is developing, whether or not it's in the following the right direction within the design space that you have come up with. So I think the answer to that question is, of course, you need to have a batch release test, but also you should start developing some of the in-process controls to allow you to better understand and better control the manufacturing process. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we've got a few more questions that came in for Dr. Antonio Costa. And here's the first question for Dr. Costa. What is the proper type of lipid selection if the API is hydrophilic or hydrophobic?
Yes, I would say there's it does not necessarily depend on the API entirely. You know, the lipid, it's a combination. So the lipid itself is more as it's going into the bilayer. So if it's more about the the structure of the bilayer that's going to form the rigidity of the bilayer, and that'll be determined by the type of lipid that's chosen. So, for example, if you have or the percentage of cholesterol in the bilayer, if you're using a DMPC lipid, which would make larger particles, versus DSPC, and then with the phase transition or the transition temperature of the lipid, that could impact potential for some of the molecules. If it's hydrophobic in the bilayer, could be released and or go into another particle or cell as it's during recirculation. So there's really other impacts, I would say, than just the formation and choosing a certain lipid based on hydrophobic or hydrophilic. But if it's more of a hydrophobic particle and it's going to be loaded in the bilayer, there may be proper selection with respect to the hydrocarbon tail length, saturated versus unsaturated. If there's interaction between the lipid and the potential or the API, but I don't believe that there's a clear indication of you have to use these types of lipids for hydrophilic and these other types of lipid for hydrophobic. It's a more complex relationship. Thank you.
Thank you for responding to that question. Here's the next question for Dr. Costa. What is the method to make the LNPS?
So the method to make the LNPs. So in general, you have your lipid, which is pre-dissolved in ethanol, and then you have your mRNA or other nucleic acid that would be in the aqueous phase. We call this we're calling this aqueous phase 1 that enters our injection port, and then essentially the LNPs are formed as these two are mixed together. Some critical factors to consider would be the concentrations, the N to P ratio. So there's different charge effects with the ionizable lipid that's present, and the nucleic acid. So you have to have a certain range for N to P of the lipid and the nucleic acid. Otherwise, you may form it may be too much of a charge or too less of a charge that's present, and then this can lead to instability of the nanoparticle. So it's really the combination of the type of lipid, concentration, N to P ratios, and then also on the process side, the flow rates. Under certain conditions, the flows are required to form a more stabilized jet, and you have to reach this stabilized jet in order to form the nanoparticles. So when you combine all of these together, you can have a nice stabilized formulation. Yeah, thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Costa, and here's the next question. Can you give us more details on how you calculate the in vivo release of DOCs?
Yeah, so this was a study that was worked on with NCL. So for any of the protocols, I would just point to their website where on cancer.gov. So they have listed multiple protocols. So NCL protocol PH-2. It outlines the methods that were used for in vivo characterization. Thank you.
Thank you for responding to that question. The final question for Dr. Costa is the following. For mRNA LNP stability, how long did you leave the formulations in ethanol?
Yeah, so for these for the cryo-TM images that we showed, so there were two different ones. One was at the 1.7% ethanol, and then the other was at the 5% ethanol. So these were left, I'd say, at least for 2 days or so, and also during a shipment to overnight to the cryo-TM facility. Once they received these particles, they then did a more of a flash freeze and stored them prior to when they put onto the scope. So it was multiple days. It wasn't just we didn't necessarily control it for hours, but that could also lead to what we see of the delamination, especially in the 5% ethanol case, and swelling. But that does come or bring us back to understanding intermediates and really understanding how long should we do transfers if we have to do a hold time or so between processing stages, which would be more of a batch approach. If you have to do a hold time, and what conditions are required and acceptable. So it's more about understanding just the the relationship between the actual nanoparticle structure and ultimately stability of these particles. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few more questions that came in for Dr. Kurt Reistroff. And here's the first question. Is the inclusion of targeting ligands on the outside of the NPs ever a complicating factor for the nanoprecipitation process? And do you get unintended interaction between these groups and the self-assembly of the core?
Thanks very much. That's a good question. Most of our targeting work has used a relatively small chemical group on the ends of those diblock copolymers. So that hexamannose moiety, for example, is not it's not going to electrostatically interact with anything. It's not going to ion pair with anything. It's hydrophilic, so it's not going to want to precipitate into the core. If you had a targeting ligand that's instead something enormous like an entire antibody, we would anticipate some potential problems there, just because if you have a diblock copolymer with a molecular weight of something like 10,000 Daltons, if you then go adding something to it that is comparable or larger in molecular weight to that polymer, then the overall diffusion of the polymer itself is going to be significantly altered, and that could potentially affect its ability to diffuse onto and absorb onto the cores of those particles during self-assembly. So we have tended to stay towards smaller ligands for that reason, and we haven't seen major issues there. Thanks.
Thank you for responding to that question. Here's the next question for Dr. Reistroff. What is the minimum volume that can be achieved with your IgM method and the IgM overall?
Good question. The smallest batches we make in the lab, we have 500 microliters of each of the two streams. I know that there is industrial interest in scaling down that mixer to even smaller minimum batch sizes. I haven't worked directly with any of the units that I think people are developing, so I can't say more about that at this time. But yeah, we're usually looking at a minimum volume of about 1 milliliter, and there is a hold-up volume inside the mixers that we use of about 100 to 200 microliters. So if we put a mill through it, we get about 800 microliters out of it. Thanks.
Thank you for responding to that question. We have one more question for Dr. Reistroff, and this will be our final question for the Q&A panel. And the question for Dr. Reistroff is the following. Can the API quality be degraded considering the high pressure involved in IgM?
That's a good question too. So the pressures are actually deceptively low. This isn't like a, this isn't a tea mixer. It's also not a high pressure homogenizer type unit. So the back pressures, I typically will drive the syringes, or my students will drive these syringes by hand at the lab scale, at the very small scale, and then when we go up to a syringe pump, we're not seeing significant back pressure. If the question is also about, well, you're going to put a biologic through this mixer, is it going to get sheared to bits inside the rapid mixing, the turbulent mixing? We also aren't seeing that. We've put enzymes through these, we've put RNAs through these, and we're not seeing physical degradation because of that shear. So we don't think that it's a major problem. Thanks.
Well, that's all the time we have for questions in this panel. A huge thank you to our speakers and panelists for answering numerous questions that came in from our audience. We're now in our lunch break until 12:45 p.m. Eastern. Enjoy your lunch. Welcome back from the lunch break. We do have a few housekeeping notes and reminders to get us started. This activity is eligible for an attendance certificate which supports continuing education by SOCA Reps, SQA, and ACRP. Please refer to our website at fda.gov/CDER-SBIA for more details. You'll be able to obtain the attendance certificate only upon completion of the survey, which will remain open for 2 weeks. Remember, this is October the 25th. Please remember to download the certificate immediately upon completing this survey. It cannot be retrieved after the fact. Let us know your comments and feedback via the survey. This will help guide the design of our future events. As a reminder, the email we send to attendees later today will have information on the survey link. Moving into our first presentation, in session 2, we'll learn about quality considerations and controls for drug products containing nanomaterials. Where are we and how we get here? By Dr. Hailing Zhang, Branch Chief in the Office of Life Cycle Drug Products, Office of Pharmaceutical Quality. Our next presentation is on advanced separations and detection and assessment of drug quality for drug products containing nanomaterials. This will be presented by Dr. William Smith, Research Scientist in the Office of Testing and Research in OPQ. And for our final presentation before the Q&A panel, this will be on our regulatory science programs and outreach by Dr. Tina Morrison, Director in the Office of Regulatory Science and Innovation in the Office of the Chief Scientist. Please join me to welcome our first presenter, Dr. Smith.
Good afternoon, everyone. I hope you enjoyed the morning session on benefit and lessons on continuous manufacturing. This afternoon's session will focus on regulatory considerations and outreach. My presentation will discuss quality considerations and control for drug products containing nanomaterials. I'm hoping to share with you some valuable experiences that we have gained by assessing and approving 2 important drug products, namely Doxil and Onpattro. Doxil was approved in 1995 by FDA as the first liposome drug product, while Onpattro is a more recent approval in 2018. Onpattro is a lipid complex injection where siRNA patisiran is encapsulated in lipid nanoparticles. I would like to start my presentation by telling you a successful story of liposome drug products and sharing with you what we have learned in the past 3 decades by working with pharmaceutical industry and academia to develop and regulate liposome drug products. The second part of my presentation will shift to a new legend, lipid nanoparticle drug products. I will end my presentation by providing you some closing thoughts for this topic. FDA has approved 12 liposome drug products since 1995. Most of them are indicated for the treatment of certain cancer. Other conditions being treated include fungal infection, pain management, etc. Most of the drug products are administered through intravenous route. Arikayce kit was recently approved in 2018 as an oral inhalation to treat certain lung disease. Generic liposome drug products development has made significant progress in past decade. With the implementation of the GDUFA user fee program, FDA is able to invest substantial resources to work closely with industry and facilitate the development and regulation of generic liposome drug products. For example, Office of Generic Drug Products has developed and published product-specific guidance, or PSG, for several liposome drug products, including doxorubicin hydrochloride liposome injection, amphotericin B liposome injection, verteporfin for injection, bupivacaine liposome injectable suspension, perfluorine lipid microsphere injectable suspension, irinotecan hydrochloride liposome injection, cytarabine and daunorubicin liposome for injection. The orchestrated effort between pharmaceutical industry and FDA has led to the approval of 5 generic submissions for doxorubicin hydrochloride liposome injection and 2 submissions for amphotericin B liposome injection. We are also working with industry on numerous pre-submission programs for many other drug products in this category. In addition to product-specific guidance, FDA has published several guidance for industry on this topic, which is summarized in the report Nanotechnology Over a Decade of Progress and Innovation, issued by the FDA Nanotechnology Task Force in July 2020. I would like to highlight 2 of the guidance which provide recommendations for human drugs. The first one is liposome drug products, chemistry, manufacturing, and controls, human pharmacokinetics, and bioavailability and labeling documentations. This guidance communicates our current thinking and provides recommendations for developing liposome drug products. The second guidance is entitled Drug Products, including biological products that contains nanomaterials, was finalized in April 2022. My colleague Dr. Olan Stephans provided a wonderful discussion on this guidance in last year's FDA NanoDay Symposium. In the interest of time, I would recommend you watching his presentation if you would like to know more about this guidance. Since the approval of Doxil in 1995, we have much better understanding of liposome drug products. Comparing to drug products with simple formulation, the quality framework of liposome drug products require more rigorous characterization program, particularly to characterize the important liposome physical-chemical properties. The characterization program of a liposome drug product should be based on the desired quality attributes to achieve the optimum clinical outcomes. In the past few decades, a great deal of research has devoted to understand how formulation design, such as the molecular structure and relative concentration of lipids, together with manufacturing process, such as drug loading method, size reduction method, can lead to the desired quality attributes. For example, high drug loading efficiency, desired and consistent liposome size and size distribution. Based on rigorous characterization program, quality control of a liposome drug product can be achieved with testing of certain properties with adequate justification. In summary, how do we achieve high quality batch-to-batch consistent liposome drug product? We heavily rely on quality by design to understand the risk in terms of how formulation and the manufacturing process would impact the drug product quality, which can lead an effective mitigation strategy. Due to the complexity of liposome drug products when it comes to characterization and testing, we expect 2 orthogonal methods to be developed when it comes to characterize some physical-chemical properties such as surface chemistry, morphology, and sometimes size and size distribution. Due to the conventional methods used in characterizing and testing liposome drug products, sometimes method validation is overlooked by sponsors. Today, I would like to take these opportunities to emphasize that we evaluate method validation from any methods in a submission with the same regulation requirements. Those requirements can be found in various guidance issued by FDA or other regulatory entities such as USP and ICH. Now I will shift the discussion to the new lipid nanoparticles, or LNPs. Lipid nanoparticles almost becomes a household term recently due to their application in the development of mRNA vaccines for COVID-19. The understanding and advancement of liposome drug product in half century has laid out a solid foundation for the speedy development of using LNPs for genetic drugs. In the review article entitled Lipid Nanoparticle Systems for Enabling Gene Therapy, published in Molecular Therapy in 2017, the author states the origins of LNP systems used for genetic drugs lie in the development of liposomal drug delivery systems for small molecule drugs. Liposome systems for delivery of small molecule drugs represent a relatively mature technology and have led to a rigorous design criteria. Many of those criteria can carry over into the design of LNP systems for delivery of genetic drugs, including size range of 100 nanometer or less, high encapsulation efficiency, circulation longevity, robust, scalable manufacturing processes, and stability. In the development of Onpattro to targeted delivery of siRNA patisiran to hepatocytes, some other specific requirements are identified. For example, in the early stage of the development, it was challenging to achieve high encapsulation efficiency. The introduction of ionizable cationic lipids provide a feasible solution. In addition, it requires neutral charge on the external membrane surface of the particles to avoid extensive adsorption of serum protein onto the lipid nanoparticles. It is also discovered that PEG lipid containing short acid chains that can dissociate from the lipid nanoparticle in vivo with relatively short half-time will result in optimum hepatocyte gene silencing potency. If we compare DOXA to Onpattro, we notice that HSPC and cholesterol are used in both formulations as a structural lipids. Both drug products include a PEG lipid derivative, but with different molecular structures. However, Onpattro also includes an ionizable cationic lipid in the formulation. Two novel lipids are used in the Onpattro formulation. First of all is the ionizable cationic lipid, D-lin MC3 DMA. During the development, a few dozens of ionizable cationic lipids were evaluated for the optimum delivery of patisiran. These lipids differ in the head groups, linkers, and hydrocarbon chain length and type. D-lin MC3 DMA is the final selection. Its molecular structure is shown here. This ionizable cationic lipid is designed to control particle formation, encapsulation of siRNA, cellular uptake, fusogenicity, and endosome release of the siRNA. Particularly, the amine group, amine pKa, is optimized to be around 6.4. It is positively charged at the acidic pH, but neutral in the blood. Therefore, at the acidic pH, positively charged amino head group can complex with the lactively charged phosphate backbone of the siRNA to facilitate association and achieve high encapsulation efficiency. Then the pH is elevated to approximately 7, so surface charge of the lipid nanoparticle is neutral in the finished drug product, resulting in a longer circulation lifetime. A novel PEG lipid is also developed for Onpattro. PEG lipids are used in liposome or lipid nanoparticle formulation to stabilize the particle by forming a protective hydrophilic layer that shields the hydrophobic lipid particle, therefore preventing the association with serum protein, leading to longer circulation time. A PEG lipid is also used to control the particle size. For example, higher levels of PEG lipid lead to a smaller lipid nanoparticle systems. In addition, modulating the alkane chain length of the PEG lipid controls the rate of PEG lipid dissociation from the particle, therefore impacting the pharmacokinetic and pharmacodynamic of the encapsulated siRNA. Comparing to carbon-18 acyl chain used in most liposome formulation, the use of PEG lipid containing carbon-14 acyl chains leads to a quick dissociation from RNP in vivo with a half-time less than 30 minutes, then resulting in optimum hepatocyte gene silencing potency. If we compare the drug loading method between Doxil and Onpattro, we notice a high encapsulation efficiency is achieved through different loading mechanisms. Doxil utilized active drug loading method by an ion gradient. Doxorubicin hydrochloride solution is incubated with liposomes where the internal aqueous compartment contains a higher concentration of ammonium sulfate. Doxil molecule is protonated in external media, then partitioning across the liposome lipid bilayer. Once Dox is in the internal aqueous compartment, it will precipitate as a doxyl sulfate crystalline, which drives more Dox molecule across the lipid bilayer, therefore to achieve the high drug loading efficiency. For Onpattro, the ionizable lipid complexes with negatively charged siRNA in an acidic environment by electrostatic interaction, which facilitate association and encapsulation. An ethanol loading process is developed to achieve high encapsulation efficiency. Manufacturing of liposome and lipid nanoparticle keeps evolving. Extrusion method is widely employed in research and early stage development. For example, extrusion is used to achieve desired particle size in the production of Doxil. Homogenization method is also developed and used to produce some liposome drug products. More recently, ethanol loading method is developed. For example, Onpattro is using a T-tube mixing process where lipid dissolved in ethanol were rapidly mixed with oligonucleotides in an aqueous buffer, resulting in efficient loading of nucleic acid into a small lipid nanoparticle systems. From the morning session, you probably also know that the same principle is used to develop a continuous manufacturing process for producing liposomes and other drug delivery systems containing nanomaterials. To facilitate the assessment and approval of drug products containing nanomaterials, FDA has invested substantial resources on method development and validation, standards development, and training in this area. In addition to research projects conducted by FDA, we have close collaborations with various stakeholders, including other regulatory entities, standards organizations, pharmaceutical industry, as well as academia. In terms of characterization and testing of Onpattro, what we have learned from liposome drug products become very valuable. morphology, surface charge, particle size and size distribution, encapsulation efficiency, and in vitro release are considered critical quality attributes and are thoroughly characterized. To close my presentation, I will try my best to discuss the question, where are we going? This figure is taken from the FDA 2020 Nanotechnology Task Force report I mentioned before. From this figure, we can see since 1970, FDA Center for Drug Evaluation and Research has received more than 600 applications, including INDs, NDAs, and ANDAs for human drug products containing nanomaterials, and half of which were submitted within the last 10 years. In these submissions, the most commonly seen drug delivery systems are liposomes, nanocrystals, emulsions. Most indications, of course, are cancer, inflammation, immune and pain condition, and also infection. And most of the drug products are for intravenous administration. The report emphasized a science-based, product-focused regulatory policy. In line with the policy, I would like to provide a few final thoughts for developing a drug product containing nanomaterials. First, quality by design should be implemented when considering the design and the intent of the nanomaterial. A risk-based approach should be adopted through the entire development and, for example, process risks should be identified with corresponding control strategy. Characterization is key through development, focusing on adequacy of analytical methods, sampling strategy, and orthogonal methods. Finally, we encourage industry to consult with the agency early in the product development. For example, pre-IND meeting for NDA submission and the pre-development meeting for ANDA submission. At last, I would like to thank many colleagues from FDA, especially the ones who helped me prepare this presentation. Saray from OLDP OPQ IQA review team, Pahala, Bing, and Andre from OLDP OPQ leadership team. Special thanks to Olan and Marie who spent their valuable time to discuss with me on this topic, especially reviewing portal and share their valuable expertise and experiences. I'm very grateful for the long-term collaboration in this area with Xiaoming, Darby, Yan, Wenlei, and Jiwen. Thank you for your attention. Please make sure if you have any questions, please ask me during the question and answer panel discussion session at the end of this session. Thank you.
Thank you. Hi, everyone. I am William C. Smith. I'm a research scientist in the Office of Testing Research here at the FDA, and I'm going to talk to you a little bit about some of the advanced analytical methods that we're using for the assessment of drug product quality for things containing nanomaterials and some of the research that we've done in that vein. The process of creating a nanomaterial containing product often goes through some fundamental steps, from the initial synthesis to the stabilization or functionality of the particles, of course the purification, and importantly to us, the characterization of those nanomaterial containing products. As we've heard from some of the other speakers, our ability to produce more and more complex nanomaterials is ever increasing with the new developments in things like continuous manufacturing. but there still seems to be a disparity between our ability to synthesize more complex products and our ability to thoroughly characterize these products. And this is because most of these nanomaterial containing products have a wide distribution in things like both the size, the chemical composition, the bulk composition, and things like surface functionality. The problem arises from the fact that many of the ensemble techniques that we are using currently are just determining the average of one such property. And that might not be sufficient for these types of products where you have multiple concurrent distributions of these properties occurring. In general, we know that nanoparticle properties fall into things like the physical, the compositional, and interfacial. And as we make more and more complex systems, like say a biotheranostic where we want to have a targeting agent, maybe an antibody, a stabilizer like PEG, some kind of tracing or imaging component, we have to be able to identify all of these individual properties well. And so as these nanomaterials are becoming more complex, how do we address these simple individual property questions? And specifically, we'll talk a little today about size distribution and morphology. Even for something as relatively simple as measuring the particle size, for a complex product, say like cyclosporin ophthalmic emulsions, the task becomes more and more difficult, and the use of a number of complementary techniques really adds to understanding exactly what we're looking at, or really identifying the whole elephant here. What I'd like to point out to you here in the bottom right hand is AF4 mols, or asymmetrical field flow fractionation with multi-angle light scattering technique. This is one of those separation techniques that allows us to not only get an average particle size, like many of these other ensemble techniques, but also to look at individual subpopulations and the concentration of particles that fall within those size ranges. Critically, the use of this multiple complementary particle sizing technique approach produced results from the research that provided pivotal support to develop a PSG and led to the approval of the first cyclosporin ophthalmic generic products. And this is also another kind of pivotal moment where this was the first time that asymmetrical field flow fractionation was used as part of a package to support bioequivalence determination for these. Today we're going to be focusing really on liposomes, but these separation techniques can really be extended to a whole host of nanoparticulate or macromolecular and polymer systems to look at things like the particle size, the molar mass, surface charge, also morphological conformation and shape. What's really nice is that we can also get this kind of secondary property information like how the drug is distributed, possible aggregation and stability information, etc. So today we're going to focus in on asymmetrical field flow fractionation, but we do have a host of other advanced separation techniques that we are investigating thoroughly in the FDA laboratories. So field flow fractionation is a class of chromatography-like analytical separation techniques that was developed first for macromolecular and colloidal separations and enables you to investigate analytes in the 1 nanometer to tens of micron size range. Also, different instrumentation is available that provides different types of external fields. So thermal, electrical, in this case we'll be talking about a flow variety, but the selection of field actually allows you to separate based on different analyte physicochemical properties. In many ways, a field flow fractionation system in the laboratory is set up fundamentally like a chromatographic system with a pump and a suite of detectors that provide you some kind of information. But the fundamental difference is between the pump and detector suite, instead of being a column, is a channel. So let's look at the mechanism here for how this might differ. In field flow fractionation, a thin ribbon-like spacer is sandwiched between two blocks through which you flow your liquid. And an external field is applied in order to cause some kind of migration of the particles. When you're flowing at these in a laminar flow condition, a parabolic flow profile is produced. And as your analyte is interacting with that perpendicular field with respect to the flow, it will accumulate next to one of the walls. This buildup in concentration is then counteracted by fixed first law of diffusion, and the translational diffusion of the particles lifts them to different mean layer thicknesses in the channel. They therefore see different average velocity profile streams inside of the system, which leads to differential elution of the analytes. When you're using an asymmetrical flow system, this is solely based on diffusional transport, and therefore you get separation by size, with the smallest particles with the highest diffusion coefficient coming out first, and the largest particles with the lower translational diffusion coefficients coming out afterwards. In general, some of the benefits of this open channel design lend themselves to low shear stress, so you're able to analyze things like loosely bound aggregates or protein coronas on interacting nanoparticle systems. You don't tend to need to filter your samples because it can handle things up into the micron size range, as you can see from the wide analyte size range. You can use a variety of carrier liquids. Because there's minimal interaction between a stationary phase, as there is no stationary phase, you tend to have limited sample loss, and you can upsize your channel to run anything from in the nanogram quantities up into the milligram quantities for some of the semi-preparative systems. So let's take a look at where we're applying this field flow fractionation technique in our current research at that. So in the following section, we're going to look at how we're using some of these field flow fractionation techniques as methods not only for determining particle size distribution, but also we're going to look at assessing their capability for determining morphological variability in liposomal drug products. So this whole project was really made possible through our collaboration with University of Connecticut, and as Tony has already talked about, their continuous manufacturing setup for producing liposomes. Really key to this whole process is that the continuous manufacturing system can produce particles with well-controlled particle size and well-controlled drug loading. Those two things in combination allow you to produce particles with really controlled morphology or shape. And this is really fundamental to our ability for assessing our analytical methodologies, as creating a well-controlled and well-defined analyte library is critical for the assessment of your methodologies. So let's put our continuous manufacturing derived liposomes from UConn into perspective here. We have an AF4 fractogram with our concentration detector intensity on the y-axis and retention time on the x-axis, our UConn sample here in the gray, and two products that we tested with two specific lots from one of the products here, and some representative TEM images for those. what we can see is that there's both polydispersity in size, you can see kind of from the breadth, but also in morphology from the distribution of both spherical and prolate ellipsoids here. Overlaid also on this is the online DLS detection. So we're getting hydrodynamic sizes for each of these particle populations across the peak as it elutes, and we can take this signal and integrate it with respect to the concentration detection for each of these specific sizes in order to produce a particle size distribution. Now, we did this integration process for a number of different samples. So here are our Yukon samples. We can see we have varying levels of polydispersity and monodispersity. And we also display this in terms of two different number and weight-based distributions, so skewed more towards the intensity or the light scattering versus skewed towards the number-based intensity from the concentration. We did this, of course, also with the commercially available products with several lots. And so what's the point of having both of these weight and number-based distributions will become evident here on the next slide. Now, what you can see here is two of our samples where we have overlaid the number and weight-based distributions for each of these samples. They have approximately equivalent Z average from batch DLS, but different polydispersities. And this becomes very evident when we look at our particle size distributions from the separation techniques, where the bigger deviation from number and weight-based distributions is exemplary of polydispersity versus a more monodisperse system where the number and weight-based distributions are nearly equivalent. Moving away from our more typical particle size distribution analysis, we can start to think about how to assess liposomal morphology or shape using our advanced separation and detection suite. And so here we just have some representative TEM images of some of the Yukon samples that were produced, where we have linear crystal morphology with spherical shape, linear crystal morphology with an elongated particle shape, or a circular or spherical particle shape with a circular doxorubicin crystal. And again, this is all produced using that continuous manufacturing process where we can create these empty liposomes and then actively load them and control that loading process in order to, in a controlled manner, make these different morphologies. Without getting too detailed, one of the ways that we're going to investigate both, the particle shape and the particle size is through a use of this online detector suite. So one, we have a multi-angle light scattering system. And what this enables us to do is to look at the intensity with respect to the angle of the light scattered for the analyte as it passes through the detector. Inside of this p theta term, we can extract the root mean squared radius or radius of gyration from the slope of this dependence. That tells us something about how the mass is distributed within the particle. And we can compare that to the DLS-based size, which is actually based on the Stokes-Einstein equation or the diffusion coefficient, which gives us our hydrodynamic size. So the spherical equivalent size for something that is diffusing through a medium. And just for clarity's sake, we're using a Berry second order for all of the data that I'm showing you here from the Malls fitting. Now we can take these two sizes from our multi-angle and our dynamic light scattering online. We can make a ratio that helps us understand something about how the mass is distributed, or the morphology, or what we call the shape factor or rho value here for our particles. So just for instance, a hollow sphere will have the majority of its mass, or its radius of gyration base size, distributed out towards the edge, if it's a perfectly uniform shell, very near the hydrodynamic radius of the particle. This is in contrast to, say, a uniformly dense sphere where there is more mass distributed towards the center than towards the hydrodynamic volume edge. And of course, we can make some theoretical predictions about what this rho value would be for things like rods or prolate spheroids, which tend to be what a lot of these doxorubicin liposomes end up looking like. Now in practice, this is what the data actually looks like, is we have our fractogram here, the same fractogram overlaid one with the hydrodynamic radius. So you can see we have equivalent hydrodynamic radiuses at these different sizes. And what's really interesting, though, is when we look at the root mean square radius or radius of gyration values, you can see that at equivalent retention time, so equivalent hydrodynamic sizes, we have differences in our root mean square radius or radius of gyration. Now, when we take those two values and make our rho ratio, what we can see is that for something that's uniformly spherical with this linear crystal, we get one specific rho value, and for one of our exemplary elongated samples, we get a different rho value. And of course, for our circular crystal morphology in a circular particle, we get, again, a different rho value. So we can take that for all those 16 different samples that we looked at, and we can kind of crunch the numbers on what those look like on average. So for our 16 samples, we ended up calculating our rho values at full width half max and averaging over each of the peaks, and then pooling these values based on the observed TDM morphologies. And we can see a trend that follows more or less with the theory that our hollow spheres end up around a rho value of 1, with a slight elevation for our elongated or prolate ellipsoid with respect to our spherical particle with a solid linear core. And so one of our questions is, when is elongation elongated enough? And so we're looking into also analyzing more rod-like liposomes in order to fit a trend for this entire series. In the end, additional studies are essential for us to really evaluate the appropriateness of this shape factor analysis as a rapid screening method, specifically in the errors involved in different light scattering models, in order to use this as a little less cumbersome method as an orthogonal method to TEM shape analysis. Again, I really want to highlight the fact that the continuous manufacturing approach really provided us the library of liposomal materials that were needed in order for us to conduct this analysis, because we had samples with very controlled sizes, shapes, and polydispersities. For anyone who is interested, there are easily locatable protocols and methods being developed for field flow fractionation for the analysis of nanomaterials, both from the NCL, the EU NCL, and from ISO, who is working on interlaboratory study to elevate this technical specification to a full international standard. Just to give you a brief look at the breadth of the standardization effort that is underway, there are a number of analytical and regulatory laboratories across the globe that are putting their weight behind the use of advanced separations like FFF for the analysis of nanomaterials like liposomes and lipid nanoparticles. Additionally, there are a set of 3 ASTM interlaboratory studies that are currently using liposomes produced by continuous manufacturing as one of the materials to be analyzed. And we think that this is kind of a key role for continuous manufacturing to provide reference materials in the future. Here I've added a truncated list of resources with a couple nice reads and a number of these different methodologies that you can peruse at your leisure. Hopefully you find these useful. Just want to take a brief second to acknowledge all my FDA collaborators and everybody who helped me work on these projects, but especially to the UConn School of Pharmacy team, Tony Costa, Dr. Burgess, and Gotham, who actually produced these particles that enabled us to start really delving into understanding these analytical procedures. And of course, I'll take any questions at the end.
Good afternoon. I am Tina Morrison, the Director of the Office of Regulatory Science and Innovation and the Office of the Chief Scientist at the Food and Drug Administration. I'm delighted to be participating in the 2023 NanoDay Symposium. I'd like to thank the organizers for inviting me to give an overview of regulatory science and outreach efforts that we do in our office, which you'll hear me refer to as ORSI. This is the office where we aim to spur innovation through creative collaborations. Before I begin, I want to orient us all just to give you a reference of where we are in the Office of the Chief Scientist. So at the FDA, we are led by the Commissioner of the FDA, Dr. Robert Califf, and he oversees not just the 6 product centers, which are the Biologics and the Drug and the Devices Center, along with the Center for Food Safety and Applied Nutrition, the Center for Tobacco Products, and the Center for Veterinary Medicine. He also oversees scores of what we call component offices in the Office of the Commissioner. Several of those that are active in the area of regulatory science are, of course, the Office of the Chief Scientist, the Office of Minority Health and Health Equity, the Office of Regulatory Affairs, the Office of Women's Health, and the Oncology Center of Excellence, just to name a few. I encourage you to look at the organizational chart of FDA if you're curious about other aspects of the Office of the Commissioner. Transitioning to the Office of the Chief Scientist, we were established in 2012 as a part of the goal for the agency to actually connect more science-minded people across the agency. Each of the centers have their own science divisions or office, but at the FDA, when we're trying to look at how to coordinate science across the agency, we're looking at ways to try to coordinate resources. The Office of the Chief Scientist is where a lot of that coordination takes place. We support the Research Foundation, the science and innovation that underpins FDA's regulatory mission. We promote scientific excellence and innovation to achieve FDA's mission. And through the National Center for Toxicological Research, we also provide research expertise and infrastructure to the FDA product centers. On the left-hand side, you can see there are 8 sub-offices to the Office of the Chief Scientist, each with their own special function. So the counterterrorism and emerging threats, scientific professional development. We oversee the scientific integrity of work across the agency, laboratory safety, technology transfer, so making sure our scientists have access to processes to protect their intellectual property, in addition to a team that works with the product centers to execute on the advisory committees. And then you have the Office of Regulatory Science and Innovation, which I'll talk with you about over the next few slides. So in ORSI, our vision is to improve and advance public health by accelerating innovations through creative collaborations that harness the best science. And our goal is to execute on programs that can deliver on that best science across FDA. We have several programs that we run in ORSI to advance regulatory science. We have 4 intramural programs and 2 extramural programs. I'm going to take a few moments to walk you through each of these programs, and while I do that, I'm also going to highlight different areas in which we've been able to utilize these programs to help advance the areas of nanotechnology and provide support to groups like Anil Patri's group in the FDA NanoCore Center, along with folks that are working in the Drug Center and the Devices Center, providing them with mechanisms to advance science that's important to them. So let's begin first with ORCI's program impact at a glance. So our office is relatively small, but we have a pretty big impact when it comes to advancing regulatory science at FDA. With respect to the projects that we help facilitate through our extramural programs, we actually work with our scientists to fund more than $100 million each year of projects through our contracts program and our grants program, which I'll talk about in a few moments. We also, through the standards program and the FDA standards executive, we support more than 800 staff who serve on 1,000 or more consensus standards committees. And I will share some highlights of this in the area of nanotechnology in a few moments. In addition to that, we have more than 100 projects that we're managing through the program called the Centers of Excellence in Regulatory Science and Innovation, and I'll share with you some updates to that program. We touch hands with more than 1,000 scientists across the agency every year when it comes to looking at and evaluating proposals, serving on technical evaluation committees, engaging on training, education, scientific workshops. We are a very busy but small, nimble team. And we are proud of the work that we do to support science at FDA. But we're not just talking about any kind of science, right? We're even talking beyond applied science and translational science. We typically think about applied science as, okay, I've had some scientific discovery, and now I want to either apply that science into developing a new technology, or maybe developing a new product, or even considering new approaches. And these technologies we can get our hands on through different tools or standards even. And then translational science, as brought to us by way of the NIH and the National Center for Advancing Translational Science, it's my perspective that they're really focusing on that bench to bedside. How do I take this science and actually bring some relevance to the lives of a patient, right? And then an FDA's role is thinking about, well, how can we do that consistently, that bench to bedside consistently for a broad range of projects and products. And in order for us to do that, we really need to harness regulatory science. And these are different methods, approaches, tools that we can use to help assess the safety, efficacy, quality, and performance of FDA-regulated products. Now, these regulatory science outcomes, if you will, would not just be used by FDA, but also by our industry and academic stakeholders who are thinking about either developing new technologies and how they can apply to FDA, or thinking about how they can develop new products and how those products would be regulated by FDA. So it's really important when we think about the scientific work that we're trying to do at the agency, our goal is to really focus on regulatory science. Okay, so that was a high-level overview of ORCI's impact and talking about regulatory science. Let's jump into a couple of these programs. So the first program I want to highlight is the OCS Intramural Grants Program. So we manage 4 FDA-wide grants programs that really allow FDA scientists who are working in novel areas of science to get additional funding outside of their current center or office. So they can apply for funding from one of the FDA Office of the Commissioner component office, as I mentioned in my introduction. That's either through the Chief Scientist Challenge Grant, where we look at novel collaborations that are cross-cutting, focusing on high-level science areas that impact one or more offices, like nanotechnology, like alternative methods. The other offices that also support grant programs is the Office of Counterterrorism and Emerging Threats through the Medical Countermeasures Grant. We also have 2 grants programs, one for the Office of Women's Health and the Office of Minority Health and Health Equity. And we help support the operation and execution of these grant programs. And when we're looking at, we take a few minutes and look at the Chief Scientist Challenge Grant Program, ORCI actually has appropriations to fund $1 million in new projects and also $1 million for second year funding for the previous year's projects. So we try to provide funding support for 2-year projects. And when we started this program more than a decade ago, we originally had 2 program areas. One in the CORES area, where CORES stands for Collaborative Opportunities for Research Excellence in Science. But this program was mainly focused on advancing nanotechnology. What we've done here, what you can see on this slide is we actually report to Congress yearly on how we've spent congressional appropriated dollars, and specifically we've had a specific call out to how we are advancing nanotechnology in support of the Office of Science and Technology Policies, what they call the National Nanotechnology Coordinating Office. And you'll see from last year's congressional justification for our intramural grants program, we actually discussed that over the last decade where we've been running this program, we've actually funded more than 70 projects in the CORES area focused on nanotechnology standards. And so I will give you a snapshot of some of those standards that have come out of the work from the nanotechnology group. So as I was talking about, the FDA standards program, the FDA standards executive works in ORSI and actually works across the agency to collaborate specifically with the medical product centers on consensus standards development. We have a database to look at standards when they're in draft format, and meaning that a standard is ready for FDA to comment on those standards. We have a standards committee that coordinates with all of the standards liaisons across the agency. An active database of current FDA standards, in particular those that are recognized by the agency to be used in regulatory submissions, and also access to the portal where we can get access to all of the standards through the different standards developing organizations. Here's a snapshot of the scope of the FDA standards program. As you can see here from each of the product centers that I mentioned before, hundreds of standards are being developed and utilized by FDA. And it's important to note that if you're not familiar with the standards program or standards development organizations, this is a great opportunity for the industry to engage with FDA and others from academia on developing methodologies and approaches that can be standardized and used to advance regulatory science. So here's a snapshot for you on the standards that are in nanotechnology. So the nanotechnology has a subcommittee that is focused specifically on consensus standards that impact the area of nanotechnology. This subcommittee has reviewed and commented on, they've reviewed at least 75 standards and commented on recently 50 draft consensus standards, and you can get access to those standards by looking at the FDA Consensus Standard Database, which is available at fda.gov. Additionally, you will find that there are 23 standards focused on nanotechnology that are recognized by the Center for Devices and Radiological Health. So if you've not looked at that database, I encourage you to check that out. And of course, standards development is always ongoing. There are always new scientific gaps and challenges to tackle. And so what I'm showing here on this slide is there are currently 7 active work items through the ASTM Standards Development Organization. There's a subcommittee called E56, and there are 7 work items that are open. So if this is an area of, if these of the standards that are listed here, if these are areas of interest for you, either to engage on, to contribute to the development of that work, or to actually learn from those experts who are developing the standards in those areas, this is a great opportunity for you to get engaged. So if you have any additional questions about standards and nanotechnology, I encourage you to reach out to either the director of the FDA NanoCore, which is Anil Patri. You've heard welcoming remarks from him this morning, and also the FDA Standards Executive, Mr. Haney Demian. So their contact information is provided here. One of the other program areas that ORSI manages is the FDA Scientific Working Groups program. And this is a very exciting place because this is one of the ways in which we can actually do cross-agency collaborations on science. We have 12 topical scientific groups. In each one of these groups, there are members from each of the FDA product centers and also members from the different component offices that I mentioned earlier. And these folks meet either monthly or quarterly to engage on communication and sharing information, scientific information, information about projects and training, also thinking about ways in which they can establish projects to tackle common goals and objectives, where we're looking at science as it applies, science at the level just before it's applied to any specific product area, for example. The other interesting aspect of bringing all these scientists together is it allows them to talk about opportunities to share resources, not just to share knowledge, but to think about ways to share infrastructure, to share laboratory space, and to think about how they might collaborate and cooperate on projects and maybe even submit projects to the intramural grants program. And what I've highlighted for you here is the nanotechnology working group, which has been called the Nanotechnology Task Force, as they have been reporting to the Office of Science and Technology Policy's larger effort in nanotechnology across the federal government. And if you want to learn more about the task force, I provided the link below. Okay, so I just gave you a snapshot of the intramural programs in ORCE. I'd like to spend some more time on the extramural programs because these are the ways in which we actually engage with our external scientific stakeholders. Before I talk about the specific programs, I want to orient you for a minute to our regulatory science framework. You know, when I've been working at the agency now for 15 years, and when you're talking about science priorities, there's always different ways in which you can communicate science priorities. You can talk about scientific priorities from a product area. You can talk about scientific priorities from a demographic or a populations perspective. You can talk about priorities from things that are emerging, public health emergencies. Obviously, COVID-19 was one of them, but also thinking about misinformation and disinformation. But we can also talk about science priorities fundamentally from the scientific areas, and we can organize them in such a way that we can think about how that science really underpins and supports FDA's mission. So if we look at regulatory science, again, I shared this definition with you earlier, which is really focusing on the ways in which we can harness science through tools or standards or qualified approaches that both the stakeholders from outside and inside can utilize to inform the regulatory process. I mentioned we can talk about science from the different product areas, right? We can look at scientific gaps by the population areas. But really what we want to do is to think about science and how the scientific gaps are actually related to the different pillars of the FDA mission. So there are three very specific charges that are related to FDA's scientific mission. The first is to think about how we utilize science to modernize the development and evaluation of FDA-regulated products. There is the science around strengthening post-market surveillance and labeling of FDA-regulated products. And last, the third charge is invigorating public health preparedness and response of FDA, but also of patients and consumers. In these three different charges, we have identified 24 areas of science that we can tackle through our extramural programs to support the agency. Now, in the short window of time I have today, I can't go through all of the details of those different science areas. Some of those cross-cutting areas are, for example, alternative methods, artificial intelligence, harnessing real-world data and utilizing complex statistical methodologies to think about real-world data becoming real-world evidence. You can find more details about the regulatory science framework on the FDA.gov website. But what does that mean for extramural programs? Well, the first of those programs I want to talk about is the broad agency announcement, or what we call the BAA program. This is where we can establish research and development contracts in a streamlined, nimble way. And the way that we do this is, the goal of this program is to really think about, we have new challenges arising every year, either on the products that we know about that we're trying to develop or new areas of science that are coming down the pipe. And FDA needs to have the ability to be nimble and to be able to adjust our scientific priorities as the science and technology changes. And so every single year, FDA publishes through a solicitation for research and development contracts. We publish every year our scientific priorities, and we lay them out in a document which you can get access to on the sam.gov website. And in that document, we've now structured our scientific priority areas utilizing the regulatory science framework and looking at ways in which science can advance each of those three charges. And this is the way that we engage with our external scientific stakeholders. So this pathway is open to both scientific folks in academia, but also in industry, whether you work for a specific manufacturer or if you're in a laboratory setting. We have a very rigorous review process, and typically I talk about the process at a high level, but we're undergoing some changes to that BAA process now. And if you are someone who is interested in the BAA program or want to hear more about the upcoming changes for fiscal year 24, I encourage you to save the date, October 25th, for the BAA Day. And the reason for that is we'll provide you with an update to some of the program and process changes that are coming. But as you're thinking about how you might apply your expertise to the BAA process, I want you to think about these three important areas. These are the criteria by which we rate the proposals that come through the BAA. And on average, we receive 250 proposals every year, and we can fund around 40 to 45 proposals. This has been our average for the last 5 or 6 years. So it's really important that we're looking for robust scientific and technical merit. We want to make sure that there's soundness and feasibility in the proposals that are presented, in addition to touching on those key scientific areas, those areas important to the agency programs, which we lay out for you in the solicitation. And last but certainly not least is the capabilities and experience. What are the qualifications of your team, and do you have demonstrated success in either other BAA proposals or contracts that have been issued to you? So this information is really important to our team to get your proposals rated highly, because we only fund those proposals that are rated high. Okay, so those related to nanotechnology. This is just a snapshot looking in the BAA database for funded projects in the area of nanoscience or related to nanotechnology or nanoparticles. You can see here that there have been several projects funded through the BAA over the last 5 years. Fiscal year 23 just closed, so we're still working on the obligated dollars amounts, but we are funding the University of Connecticut again. You guys will hear from Nicholas today and his presentation around the work that they're doing at the University of Connecticut in the area of nanotechnology. And actually, I believe there's a presentation from Diane Burgess on the continuous manufacturing of liposomes and LNPs. While this slide is only showing one of the contracts, it is my understanding that our drug center has actually awarded over the years about $5 million to the University of Connecticut, either through the BAs, but also through our CERC program, which I'll talk about momentarily. And also want to point out that last year, thanks to the America's Job Recovery Act, we were able to fund a BAA from the Center for Biologics. We awarded an $82 million contract to MIT on continuous manufacturing of mRNA. And so we're really delighted that we were able to take that funding and put it into an institution to advance the areas of continuous manufacturing system. Okay, so now I'm going to transition to our Cooperative Agreement Grant Program, or the Centers of Excellence in Regulatory Science and Innovation, or what we call the CERCs. The CERCs are academic institutions that we work with to establish regulatory science projects. We do have a request for proposals for different project areas. That being said, this mechanism for engaging with external stakeholders is different than the BAA in in that for each of the projects, we have both CERSI collaborators and FDA scientists on each of those projects. And a bulk of the work that we do through the CERSI program is through our regulatory science projects, but we also, the CERSIs also work on developing workshops and lectures, training courses. We have scores of fellowship and scholarship opportunities. We have a regulatory science competition that we put on every year. So there are a lot of different ways in which the CERSIs work alongside FDA to advance regulatory science. And I'm proud to announce that we actually have a new CERSI institution that we've inducted into the program this year. We just started our new grant cycle on September 1, 2023. A typical cooperative agreement grant cycle lasts for 5 years. Our newest CERSI, the Triangle CERSI, is a collaboration between the University of North Carolina at Chapel Hill with Duke University, NC State University as well. We had a very competitive new funding opportunity that we established last year. We had almost 30 applications for establishing a new institution. So it was a very strong competitive process, and we're really excited for the 4 CERSIs that have been working with us the last grant cycle to be renewed, and also to bring a new CERSI on board. I really encourage you to visit the FDA.gov website to learn more about each of the specific institutions and the scope of work that they are doing to support regulatory science at FDA. One of the things I just want to highlight for our program is that we've established what we call outcomes of interest. Because the bulk of the work that we do through the CERSI program are regulatory science projects, and at this time we have more than 100 projects that are ongoing, and in total for the decade that we've been running this program, we've had almost 250 CERSI projects. So that's quite a lot, and we're trying to think about what's the impact of all those projects. So when we think about establishing a project with our goal to advance regulatory science, our ultimate goal at the end of the day, if we think of that blue arrow at the bottom of the screen as time, is our hope is that by taking these projects and sharing the knowledge, that will catalyze some action in the community, either to get more funding for a particular area of science or to establish a new program or effort, ultimately getting to the place of finding some tangible outcome that can allow us to inform regulatory decision making. And so on this slide, I'm giving you a snapshot of information that is detailed on our website, but it's the very specific activities we're looking to show impact for our regulatory science projects. And so beyond the disseminating of scientific knowledge through peer-reviewed publications, through presentations and training, we're also looking to further action, which we know can take time. If you've ever done a scientific project, you know that getting to a place where you're catalyzing action can take a few years. And so thinking about, what are some ways in which these projects that we're funding can actually go to the next level and maybe end up as a qualified tool, a consensus standard, a reference material, something that we can actually use in a guidance document that can be used to inform regulatory decision making, both for FDA and our scientific stakeholders in industry and academia. So I encourage you to take a look at these outcomes of interest. And so with that, as I'm coming to a close of my presentation, I just want to take a moment to highlight a project, a CERSI project that's focused on nanotechnology. And this project comes out of the University of Maryland CERSI, and this project is titled Hyperspectral Interference Scattering Microscopy for Characterizing Nanoparticle-Based Therapeutics. The PI from the University of Maryland is Dr. Taylor Wohl, and we have several FDA SMEs and collaborators on this project from the Center for Drug Evaluation Research. The main impact of this work is to advance regulatory science knowledge, meaning to gain and to elevate and increase the scientific knowledge of the FDA regulatory teams. The work achieves this impact by developing a new approach to rapidly characterize nanoparticle-based drugs, which can be used by the pharmaceutical industry to more completely characterize those drugs. And in particular, the goal is to develop generic nanoparticle-based drugs. That requires rigorous chemical comparison to the name brand originator drug. Currently, there is a lack of analytical tools to fully characterize nanoparticles in drugs, making it difficult for generic drug developers to apply for FDA approval with sufficient data. So this technique helps to utilize simple sample preparation, small volume batches, simple data acquisition, and has the potential to be made into a high throughput technique. The first outcome from this project has been a publication which came out recently, and the team is working actively to undergo the establishment of 2 additional manuscripts, and Dr. Wool actually presented these findings at a recent meeting in Portland. So we're very excited. If you want to read more details about the project summary, you can scan the QR code that will take you to that page. All right. So in summary today, you heard me give an overview of the different ways in which ORCI is trying to establish creative collaborations with our external scientific partners to harness the best science. We have many programs that connect our scientists across the agency on a myriad of scientific topics, including nanotechnology. We have a small but nimble team to support FDA scientists and their collaborators, and these are the faces of the folks behind the scenes. So thank you very much for the opportunity to share with you our regulatory science outreach efforts, and please feel free to contact me if you have any questions or follow-up comments. Thank you so much.
Thank you for the great presentations. We'll now enter our final Q&A panel. As a reminder, if you haven't entered your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. We do have some few questions coming in right now. The first group of questions will be addressed to Dr. Haling Zhang, and here's the first question for Dr. Zhang. Most of the approved liposomal drug products are administered via intravenous route. What is the rationale? Thanks.
Thank you for that question. Most approved liposomal drug products are either indicated for certain cancer or pain management. Therefore, other routes such as oral, it's not feasible in terms of the bioavailability. And also, most approved liposomal drug products are within around like 100 nanometer range, which is also make oral administration challenging in terms of the bioavailability. Thank you.
Thank you for responding to that question. We've got a few more questions that came in for Dr. Zhang, and here's the next question. What is the half-life of lipid particles in blood pH?
Thank you for that question. I think it depends on which drugs we're talking about. In terms of Onpattro, I believe the half-life is around 3 days. But for example, doxorubicin, it's about a day, 28 hours to 40-something hours. So it really depends on which drugs we're talking about. Thank you.
Thank you for responding to that question. The next question for Dr. Zhang, can you provide examples of acceptance criteria for encapsulation efficiency and comment on how critical this metric is for FDA? Also, what is more important, high efficiency, repeatability, or both?
That's a great question. Thank you very much for asking that. So when we're talking about the control, quality control in terms of encapsulation efficiency, we usually use the encapsulated drug versus a free drug as a testing to control this particular property. It depends on what drug we're looking at. For example, doxorubicin, we usually look at 97% of encapsulated drug. And for example, Ambusem, we're talking about usually 97 plus percentage of encapsulated drug. The rationale behind it is that for both of these two liposome drug product examples, the free drug usually will lead to toxicity. So we try to minimize the percentage of free drug in the finished product. In terms of the encapsulation efficiency, which is defined as the drug being encapsulated divided by the drug added to the formulation. If we talk about that concept, it usually will be decided during the development, depending on the clinical outcome. And usually a high efficiency usually means to actually high reproducibility too. Because when we figure out a way to achieve high efficiency, we usually able to reproduce the result, have a better process control, because you end up losing less drug during the process. So I think if high efficiency and the repeatability actually are somehow correlated if we're looking from the manufacturing process perspective. Thank you.
Thank you for responding to that question. We have one more question for Dr. Zhang in this round, and here's the question. Could you please state the allowed percentage of plus or minus in certain particle size and PDI for LNPs for successful reproducibility testing?
Thank you for that question. So in the regulatory perspective, from regular perspective in the world of testing, when we talking about the particle size, so I like to use particle size and the size distribution, because in most cases we are looking for a distribution control instead of average particle size. So for most of the drug product containing nanomaterials in the nano range, we are usually looking for D10, D50, D90 control with a certain range. As I mentioned, those quality attributes or their acceptance criteria will depend on how it is used in clinic, and also how the formulation and manufacturing process well controlled to lead the consistent particle size and the size distribution. So we don't really have a percentage-wise like plus or minus. We look at a range, and this range for example D10, D90, D50, D90 will depend on specific drug product we're talking about. Thank you.
Thank you for responding to that question. We'd also like to welcome to our panel, welcoming back Dr. Neil Patry, and introduce to the Q&A panel Dr. Olin Stevens, chemist in the Office of New Drug Products and OPQ. Moving on to our next panelist, we do have a few questions that came in for Dr. William Smith, and here is the first question for Dr. Smith. Is FFF, 3 Fs, a technique accepted by FDA for DP characterization?
Thanks, Jeff. Yeah, so in general, as we go into the nanomaterial containing products or complex drug products, we see in applications a number of kind of novel or innovative analytical approaches. So any proposed analytical method by an applicant is well considered. And kind of to Tina's point previously, because these things are so complex and we still are in that kind of area where we really need good analytical techniques for characterizing these products, there is a lot of opportunity for using kind of some of these unique separations techniques or novel analytical methods. FFF has been used for years and years in polymer and macromolecular fields. And I think in general, there's a lot of room for improvement in product characterization in pharmaceuticals and for the development of some of these kind of more novel techniques going forward. Thanks, Jeff.
Thank you for responding to that question. The next question for Dr. Smith is the following. What do you mean by orthogonal methods?
Okay, that is a good question. So in general, when we're measuring any material, or in this case, nanomaterials, we tend to kind of group these different techniques into kind of either orthogonal or complementary methods. And so in general, an orthogonal technique is going to be measuring the same, what we say is like the same primary measurand. So like say particle size. And so an orthogonal technique would, they both be measuring particle size, but they will use different physical principles for the measurement. Say cryo-TEM is looking at a projected image, so it's measuring usually like the ferrite diameter of a particle versus something like DLS, where dynamic light scattering is measuring primarily the diffusion coefficient and calculating particle size. So they both are measuring particle size. They're using completely different physical principles. Therefore, they're orthogonal to each other. Now, a complementary method would be something where you're measuring the same primary measurand, but you are also using some kind of similar principle. So say like nano tracking analysis is also measuring the diffusion coefficient. You're looking at the same sample, you're getting two particle sizes based on diffusion coefficient. Those two methods are complementary. So in general, it's nice to kind of have a suite of both complementary and orthogonal techniques when evaluating really complex types of samples. Hopefully that didn't get too semantic, but that's what we mean. Thanks, Jeff.
Thank you for responding to that question. As a follow-up to this question you just answered, Dr. Smith, which method or equipment is recommended for measuring 50 nanometer sized nanoparticle concentration in terms of particles per milliliter? And is it DLS based or an NTA based method?
Okay, so that is another good question. It is, in general, for nanomaterials, it is very difficult to get total particle concentration. So things like NTA are often used because they directly are assessing or measuring individual particles. That would tend to be one of the ways. If you're in that 40 to 50 nanometers and above, sometimes you can also use resistive pulse sensing to do individual particle counts. But in general, you never would use DLS because it's just measuring the intensity of light scattered. So for particle per mil counts, there are fewer ways than for, say, micron particles to get that value. But those would be two ways that you could do it. Thank you. And I realize now it's Ray, not Jeff. Sorry, Ray.
Thank you for responding to that question. And one final question for Dr. Smith in this round. And here's the question. It's regarding unencapsulated drug product and how is it removed from nanoparticles? And what techniques were used to determine the purity of the nanoparticles?
Well, that is an interesting question. In general, at least for the systems that we were talking about, you have the loading, as Tony described, is an active loading process where you have two streams of your empty liposomes and your say your doxyl hydrochloride, and there's an active mixing. They're in contact for some period of time to get drug inside the particle. And the beauty of the continuous manufacturing is that after that, there's another tangential flow filtration step, which basically acts as a diafiltration step to remove any excess free drug. So in general, you can think about, at the end of the process, all of the drug that is present there should be encapsulated as the free drug tends to be removed. As far as purity is concerned, if you're talking drug distribution between encapsulated and non-encapsulated, it really depends on the product as far as whether or not you can do it with something in situ, like say a UV vis shift in the peak to show that the encapsulated form has a different signal than the free form, or you can do some kind of preparative separation like diafiltration after the fact to measure the purity. I think those are kind of the two best ways. Yeah, thanks, Ray.
Thank you for responding to that group of questions. Moving on to our next panelist, we have a group of questions that came in for Dr. Tina Morrison. And here's the first question for Dr. Morrison. So if one final product is found to be successful out of one of your science collaboration programs, who will then be the owner of this product? And who will be the third party to validate and investigate further details for final approval?
Yeah, thank you so much for that question. And I think it really depends on the program. So I want to highlight an example from the BAA program. So you all heard from Antonia Kostia's group at the University of Connecticut. They received funding through our BAA contract, and they are the owners of the intellectual property there. And I believe, if I'm remembering correctly, he started a small business that has now extended that technology. And so there are opportunities through the contract mechanism for the developers of the technology to still maintain the rights to the property. In fact, if you all visit the FDA.gov website and you look for the BAA Day from last year, 2022, there was a presentation from Dr. Alice Welch, who talked about the details of the intellectual property as a part of the research and development contracts mechanism. Now, when it comes to scientific collaborations through the CERC program, again, these are cooperative agreement grants, and typically the IP stays with the investigators at the university. Now, to be more specific in terms of the different regulatory science outcomes that could impact, say, the regulatory process, those methodologies could be in the form of consensus standards, which those are actually belong to the community and can be referenced and utilized by anyone in the community. One of the ways in which FDA would get involved again in some kind of method or technique would be through our tools development programs. That's either part of the medical device development tools program or the drug development tools program. And so any independent organization can present those tools, those methodologies to FDA and work to receive qualification where FDA qualifies a very specific context of use for how those tools can be utilized in the regulatory process, either to support industry stakeholders or FDA in their evaluation of the different products. The last thing that I wanted to share with respect to the IP in any of these scientific collaborations is that those IP discussions always happen up front. So depending on if the group is targeting a specific method or approach, it's really important that those conversations happen early between the external scientific stakeholders and FDA.
Thank you for responding to that question, Dr. Morrison. We do have a few more questions for you, and here's the next question. How often does the FDA Scientific Working Group report out to the public, and where can we get the results from the FDA's current thinking on new methods, biomarkers, or toxicology?
That's a terrific question. A lot of the FDA the activities that come from the scientific working group, if the goal is to have an outward facing communication to the public or to our external stakeholders, all of those details would be available on our fda.gov website. So a couple examples of some reports that have come out of the scientific working groups have been a recent report on the update. There's an alternative methods report that came out in 2022. And recently there has been some continuation of that discussion with a new alternative methods program. So there's a new website that communicates all the relevant information there. There has been a report from the Modeling and Simulation Working Group that came out in 2022, I believe in the fall, on successes and opportunities for modeling and simulation in the variety of different FDA products. There's also a predictive toxicology roadmap that the Toxicology Working Group prepared, and that roadmap is also on the fda.gov website. So we do our best when we are preparing reports or white papers that we put those on our fda.gov website. Now, there was also this past spring a workshop on the biomarkers program, and the biomarkers program has actually put together a scientific publication. And we try to make those publications also available on our fda.gov website. And lastly, just to point out, while you can't find specific information just yet on the fda.gov website about the scientific working groups, we are working to build websites to communicate in a more transparent way about the activities of the different scientific working groups. And so stay tuned for the showcase of the FDA website for those hopefully coming in the next month. So thank you for the question.
Thank you for responding, Dr. Morrison. We have a next question just came in, and here's the question. Is there no cooperative agreement grant with the VCU or international FDA or academia?
Thanks for the question, Ray. Before I jump to the question about the cooperative agreement grants, I did want to just share with you, since I mentioned the tools program, both from the Drug Center and the Device Center, what I wanted to point out is when a tool is qualified, those details around the qualification are available on FDA's website as well. So coming back to the question about a cooperative agreement grant with VCU, I'm going to assume that means Virginia Commonwealth University. And I don't believe we have a specific agreement with them that I'm aware of through our CERSI program. That being said, we have several other cooperative agreement grants with different institutions that are sponsored by specific centers. So for example, our Center for Food Safety and Applied Nutrition runs a CERSI program where they have 4 institutions that focus on regulatory science projects geared towards methodologies for food safety. Now, that is not part of the CERSI program that's run out of the Office of the Chief Scientist, so that's why you didn't hear me speak about that. Additionally, the Center for Drug Evaluation and Research also hosts a myriad of cooperative agreement grants, and I actually believe there was one stood up earlier this year focusing on continuous manufacturing. So I would encourage you to look for those other cooperative agreement grants through the fda.gov website. Lastly, I'm going to interpret international FDA to mean agreements with other regulatory bodies. And the short answer is yes, we do have other types of cooperative arrangements with regulatory bodies. When it comes to the regulatory process specifically for drugs, there is the International Committee on Harmonization, or ICH, that puts out a lot of guidelines that can be utilized across different regulatory bodies like the EMA and the PMDA in Japan. Additionally, for the medical device space, there is the IMDRF, which is the International Medical Device Regulators Forum. In addition to those committees that focus on aspects of the regulatory process, the Office of the Chief Scientist is also pursuing opportunities for these regulatory bodies to connect on areas of science together. So in a way to consider how we might be able to harmonize on different regulatory science topics, such as some of the ones I mentioned with respect to tools. And the alternative methods area is very ripe for us to think about how we might be able to collaborate scientifically with some of our counterparts at different regulatory institutions. And of course, we have, as you heard from my presentation, a myriad of different agreements with different universities. And we also have one last note about our engagement activities. You can look on FDA.gov's website for what we call Memorandum of Understanding. And there's hundreds of these MOUs that we put together with a variety of different organizations, including academia, where if we're trying to work on a specific initiative together, we outline the scope of that initiative and who the partners are as a part of that. So I encourage you to check out FDA's Memorandum of Understandings, and you can find national and international MOUs on that website.
Thank you for responding to that question. We do have one more question for Dr. Morrison. It's a two-part question. We'll pose a question to Dr. Morrison first, and then if Dr. Morrison can pass over to Dr. Olin Stevens for a follow-up response. And here is the question. How come there's no special program for cancer treatment while there is a special program for tobacco products?
Yeah, thank you for that. So there are actually a special program, just to utilize the same language as the person who asked the question. What I will say in the overview of the FDA, there are product centers, and those centers focus on the specific regulatory aspects of products. So yes, there is a tobacco center. That being said, the FDA acknowledges a key role for coordinating efforts across the medical product centers. That would be the drug center, the biologics center, and the medical device center. There's a coordinated effort through what's called the oncology center, which is one of the component offices as a part of the Office of the Commissioner. And that office does in fact focus on a variety of devices and drugs and methodologies for clinical trials specifically geared towards cancer treatment. Dr. Stevens, do you have some additional remarks to add?
Oh, sorry, there it goes. Yeah, thank you for passing it over. I actually am a reviewer that supports the Oncology Center for Excellence. And I believe it was 2 years ago that the SBIA ran their own 2-day seminar with the Oncology Center of Excellence. So I'd refer you back to those programs that were discussed 2 years ago on SBIA's website. And as Dr. Morrison mentioned, there are a number of programs that are specifically geared on the review side towards promoting products for oncology. One of them is Project Catalyst, which fosters early stage product innovation, and another related one called the Oncology Regulatory Expertise and Early Guidance. And this is targeted at product type advice that comes in even before a pre-IND meeting package comes in. So there are active programs that reach out and help to address regulatory issues for early stage product development before they would come in for an application to prevent them from immediately going on hold and to help them in their own development. So thank you for the question.
Thank you for responding to that question. We'd like to bring Dr. Morrison back in to provide some follow-up statements on cancer treatments. Dr. Morrison?
Yeah, thanks again. I just wanted to highlight, as I mentioned in my overview of the regulatory science framework, that the regulatory science framework highlights areas of science that can impact multiple centers, can impact projects, different product areas, or different demographics and populations. What I want to point out is for those of you working in that space of oncology, I encourage you to look at the upcoming BAA solicitation it's in the details of the solicitation that you'll be able to find the most imminent science priorities for all of the centers. And in particular, you will find several key priority areas from the Oncology Center of Excellence. So there is a mechanism currently available for you to get more information on what are some of the science priorities related to oncology and cancer treatments.
Thank you for responding to that question. Moving up to the beginning of our panel, we've got a question that just came in for Dr. William Smith. And here is the question. Is it possible to incorporate hydrodynamic size measurement techniques like FFF, DLS, or NTA in line with continuous measurement so that the aliquot of the product is measured as soon as the synthesis is completed and a real-time information is obtained during the synthesis process?
Okay, hold on. Can you hear me now? Okay, great. Yeah, that's an interesting question. So in general, because of how the measurement principles of both FFF and NTA work, you could do those technically like at line. You wouldn't be able to do those in an online fashion. As opposed to the dynamic light scattering, there's been a really recent and interesting development that Tony Costa talked about in his talk, which is using DLS as an online PAT system. And so that is using what is called spatially resolved dynamic light scattering. And so that has a number of advantages, despite also being able to measure things at flow conditions, so at high flow rates, but also in relatively turbid systems. So if you are really interested, I'd go back and look at some of Tony's papers, or you can look up specifically spatially resolved dynamic light scattering, and that is currently being used online on nano continuous manufacturing systems to provide that real-time feedback about particle size for those systems. Thank you.
Thank you, Dr. Smith, for responding to that question. We do have a couple more questions that just came in for you, and here's the next question. You mentioned the use of orthogonal particle size sizing techniques. Why would someone use one of these separation techniques over a quicker or simpler method like batch DLS?
Oh, thanks for that also. I think in general, there is something to be said for the ubiquity and use of things like DLS. They are simple and quick. But again, the resolution capability and the fact that you're looking at an ensemble of every particle that's in the system, and those tend to be the sizes you get tend to be skewed to the larger size range due to the intensity-based nature of the measurement phenomena, lends itself to the fact that you might want, if you see some kind of polydispersity in your sample, to go to a separation-based technique, because so like in TEM, you might be looking at 100 to 1,000 particles. In a separation technique, you're looking at on the order of like 10 to the 8 to 10 to the 10 number of particles. And it really helps you to identify those particle subpopulations that are leading to that polydispersity. And with something like an FFF approach, you can also collect those for subsequent offline analysis. And there's some really good work by the Nanometrology Institute in Japan, where they demonstrated that if you separate a sample, fractionate it, and do TEM in order to get a really definitive particle size distribution while coupling to transmission electron microscope, you only really need to measure between 20 to 100 particles per image to get a really nice look at the particle sample distribution. So the combination of the number of particles you're looking at, the subpopulations you can identify, and then complementary offline or orthogonal offline analysis is just indispensable for looking at the whole picture of what's inside one of these complex nanomaterial containing products. Thanks, Ray.
Thank you for responding to that question. We have one more question for Dr. Smith, and it's the following. You discussed the use of shape factor analysis for liposomal products, but are there other products that this could be applied to?
Yeah, so this is another great question. In general, because of how the shape factor analysis actually works, you're kind of looking at a mass distribution inside of your analyte. So there's been a couple recent publications, I think, from both NCL, NIST, and Fanny Caputo and Jeremy Perot, where they looked at the loading, so different mass loadings of mRNA or RNA into lipid nanoparticles, and you could use that shape factor analysis to look at something as kind of a metric for RNA loading. I know they also use this in polymer micelles and a number of other things. So I see this being used in further nanomaterial characterization in the future for all of those types of products. Thank you.
Thank you for responding to that group of questions. Moving up to the beginning of our panelists, we've got a few questions that came in for Dr. Hailing Zhang. And here is the first question for Dr. Zhang. In addition to Doxil, FDA has also approved many other liposomal drug products. Can you highlight some of the differences of other liposome drug products in terms of quality considerations?
Thank you for that question. So yes, we, in addition to Doxil, FDA also approved quite a few liposome drug products. And some of the drug products actually have a different morphology from Doxil. Therefore, they could have a different quality attributes when we look at the quality. For example, if we look at amphotericin B liposome products, it's a liposome drug product where the amphotericin B is embedded within the lipid bilayer without PEG. So obviously, when we look at the quality characterization, quality consideration and control for ambisome, we look at different properties. So that's just one example I would like to highlight. So I guess depending on the design and also the intent of the drug product, the quality attributes, the critical quality attributes characterization testing will be very different from one to another. So quality by design and the risk-based approach are always critical in terms of development and also regulatory assessment and approval of a drug product containing nanomaterials. Thank you, Ray.
Thank you for responding to that question. We've got another question for Dr. Zhang, and here's the question. There are no quality guidances specifically for lipid nanoparticle drug products. Do you have any recommendations in terms of what guidance industry should follow when developing lipid nanoparticle drug products?
Thank you for that question. It's true that the agency has not published any specific quality guidance for drug products using a lipid nanoparticle as a drug carrier. However, due to the similarity between liposome drug products and lipid nanoparticle drug products, lots of principles highlighted in the liposome drug product guidance I mentioned in my presentation. It's highly applicable. I think this is also noticed by industry. We do see the industry follow the guidance when they develop drug products using lipid nanoparticle as the drug carrier. So that's one guidance I would recommend. Another one is the one I already also mentioned in the presentation, which is the newly published guidance for drug products, including biologics containing nanomaterials. This guidance contains some high-level recommendations, including the quality consideration for the product in that category. Thank you, Ray.
Thank you for responding to that question. We have one more question for Dr. Zane regarding generic drug product development, and what are the specific challenges for developing a generic drug product containing nanomaterials?
That's a great question. Thank you very much. And also a relevant question from the audience asking if there are any generic version of Daxo on the U.S. market. I think I mentioned in my presentation, we approved 5 ANDA submissions for doxorubicin hydrochloride liposome injection between 2013 till now. We still have a few in our pipeline. So there are some unique challenges, when coming to developing and assess and approve generic drug product. The biggest challenge I would like to say is the establish the equivalency between the ROD and the old reference drug product and the testing generic drug product, especially with regard to the liposomal physical chemical properties, because of the complexity of the formulation and also manufacturing process. Another challenge I would like to highlight, I think Xiaomi and other speakers also highlight, is that the characterization in terms of the analytical method validation, and also data interpretation. So we tackled this challenge from the totality of the data. So when we look at a generic submission for a drug product containing nanomaterials, for example, liposomal drug product, we were not rely on one single characterization testing. We usually rely on the full picture, the development program, how the risk being mitigated during the development to establish the equivalency between the RS, the reference product, versus the testing generic product. Another challenge is the drug product release and testing. What should be included, what should not be included. We also look at this challenge from a really the totality perspective. For example, we usually asking morphology testing be included, but lots of the morphology testing also using has to use very unconventional method. For example, cryo-TEM. We do acknowledge the challenging using cryo-TEM as a QA tool, quality assurance or quality control tool. So we will look at the history of the development program submitted by the sponsor, and sometimes negotiate and have agreement with the industry in terms of some really complex testing. How frequent should be tested, how many samples should be tested. So in that way, we do have some level of flexibility with regarding to the testing. I hope I answered the questions. Thank you, Ray.
Thank you for responding to that question. We've got just over a couple of minutes left. Moving on to Dr. Tina Morrison, we have a question that just came in, and hopefully we can get this answered. And here is the question for Dr. Morrison. Can you describe how the BAA contracts differ from the CERSI grants?
Thank you so much. So it's my understanding the audience participating in this workshop today is mainly our colleagues from the external scientific community. And there are two different ways I discussed that we can engage with our collaborators on different projects. Now, if you are interested in a CERSI project, it's really critical that you connect with those 5 different institutions, a PI at those institutions, and connect with them on ways in which you might be able to establish a project with them and potentially with the FDA. So we are not able to directly give funding to our external collaborators outside of the CERSI program through the CERSI grants. Our funding for that grant program goes directly to those institutions for a specific project. Now, we have with the BAA contracts, this is an opportunity where on a yearly basis, we put out a call or a request for scientific proposals to address the myriad of scientific priorities that FDA has. And as I mentioned in the response to one of my questions earlier, we publish an annual solicitation on the sam.gov website. That's sam.gov. You can search for the BAA, and you're welcome to submit a proposal to the FDA to demonstrate the technical merit of the work that you're doing, the team that you have, and the approach of the project that you think can actually support FDA. And then FDA would fund you directly for that work and would work alongside to ensure deliverables and milestones are met. So one mechanism is directly to the offerers, is what we call them, and the other mechanism is to the academic institutions by way of connecting with those investigators. Thank you so much.
Well, that's all the time we have for questions. In our final panel, a huge thank you to our speakers and panelists for answering numerous questions that came in from our audience. For our symposium closing, we'd like to welcome back Dr. Olin Stevens to provide some closing remarks. Welcome, Dr. Stevens.
Thanks, Ray. Well, we've made it through an entire day of talks and vigorous Q&A. I wanted to thank our speakers, our behind-the-scenes technical support, and especially you, our participating registrants. Your interest in this field makes this event possible. Your questions help drive our research, policy development, and future symposia. I hope that we can all continue to, quote, learn from the future, as Zhao Ming suggested, so we can learn from current trends and your questions to anticipate the future, and maybe positively influence that future. Hopefully the talks today help the nanomaterial field to make future products that are accessible in the future, allow industry to remain agile for an uncertain future, facilitate individualized medicines that are affordable, and engender the public's trust in these future therapeutics. Whether we impact the future by early engagement with the emerging technology team or by in-process PAT controls that look into the future quality of the product, or through understanding the potential of some of the new manufacturing processes that we heard about, maybe we all got a glimpse into that future. Again, the technical depth of your questions received were impressive and helpful. We will take the questions we fielded today, as well as those we were unable to answer, into consideration for our future regulatory actions and communications. Thank you for this feedback. I wanted to acknowledge Dr. Zhu for his leadership in organizing this symposium with this SBIA and their network for helping connect all of you. So I thank you for your participation today, and hopefully the symposium has influenced your participation in future NanoDay events. Ray, can you close us out with your admin updates?
Thank you, Dr. Stevens. And we do have a few closing reminders. Please remember the 14-day deadline, that's October the 25th, for the certificate of attendance to support CEs for SOCR, RAPS, SQA, and ACRP. Please remember to download the certificate immediately upon creating the certificate. Otherwise, this opportunity will be lost. Recordings will be posted in approximately 7 to 10 business days. On behalf of CDER Small Business and Industry Assistance, we hope that you enjoyed today's presentation and found them helpful. We look forward to your presence at our next SBIA event. Thank you for attending, and this concludes today's conference.